My name is Paul Craig. I'm a professor in the School of Chemistry and Materials Science at the Rochester Institute of Technology in Rochester, New York. And I'm part of a team that's presenting our crash course for today. I'm going to share my screen and go through a few slides, and then I'll turn it over to one of the other presenters. Right, so our crash course today is CodeBMB, Computational Literacy for Biochemistry and Molecular Biology Education. And this is based on a NSF-funded grant that I share with 3 other colleagues, and I'll show you their pictures in just a minute. And our goal here is to help people get started with coding and get comfortable with using coding and AI. And so during the workshop, or the crash course today, we do have an agenda, and I'll show you the agenda in a minute. But our primary agenda and goal here is to help you get comfortable with enough with this material that you're going to be starting to use it in your research and in your teaching. And the workshop will run for about 4 hours. So just some general instructions, and this is a summary of an email that went out to every participant who signed up earlier this week a couple of times. The best way to do this is from a Gmail account. That will enable you to save notebooks into your Google Drive. You don't need a GitHub account to participate in the crash course, but you are encouraged to set one up so that you can participate more in our community. Google Chrome is the best browser to use with Colab. Google Chrome is a Google product, and Colab is also a Google product, so it's native. But I believe that Chrome works in other browsers, Safari, Firefox, and so forth. We will be using 4 notebooks for today's workshop, and so they're numbered here consecutively, 0 through 4. You received links to those notebooks in the email you got earlier this week, and in addition, whenever we go to a part of the workshop where we will be working on a notebook, we'll put a link to that notebook so that you can see it and click on it and get there if you don't already have that link. And let me introduce the team. Team consists of Chris Berndsen from James Madison University, myself from RIT, Mike Foster from Penn State, and Wally Novak from Wabash College. Mike won't be joining us today. But Chris and Wally will be leading sessions later today, as well as myself. Here's a summary of our agenda. So after the introduction, Wally's going to give a presentation about the pedagogical basis of coding, and then he's going to talk for a bit about GitHub and forking repositories and just using GitHub in general. And we really don't know exactly the timing of how things are going to work here, but we're going to stay with our schedule. So if we finish something early, we'll take some time where people can ask us questions. We can work on things. And there is a Q&A option for all participants in the workshop. And so if you have any questions, or if you encounter a difficulty, if a notebook won't open for you, for example, put something in the question and answer tab, and we'll address that. After Wally, then Chris Berndsen is going to talk about, introduce you to Colab and Google Drive and how we're going to be using that for today's crash course, but also for other workshops that are part of the PyBMB project. We'll take a break at 2:30 for about 15 minutes. We'll come back from that at 2:45, and I'll be talking about the basics of Python syntax. We'll be looking at variables, loops, logic, lists, some other things, and there's a Colab notebook for that. We'll take another break, then we'll come back, and Chris is going to take us through Pandas and plotting. Pandas is a library in Python that enables you to work with 2-dimensional arrays in a very, very nice ways. It's kind of a Python version of a spreadsheet, but it's much more than that. We'll take another break, and then finally, Wally's going to lead a session about linear regression. And we'll be looking actually at a standard curve for protein calibration. At the end, at 4:45 or so, we'll have a recap. And also, that's time for the exit survey. And that's really important for us. We want your feedback.
Welcome, everyone. We're glad you're here. Before we touch even a single line of code today, I want to spend about 20 minutes talking about why we're doing this. And I hope that this will be more interesting than it sounds, mostly because I'm going to show you some data that I hope will make you feel a lot better about where you're starting from. So my name is Wally Novak. I've been teaching at Wabash College for 17 years, and I've been coding in biochemical research for almost 3 decades and thinking about coding in biochemistry and molecular biology education for maybe the last 5 or 6 years. Despite coding for almost 30 years, I've never coded enough to be an expert coder. Whenever I start a new coding project, I think of the algorithm that I needed to develop, but then I spend a lot of time looking up the exact syntax of all the different lines of code. And that's fine. In fact, it's great. And that's kind of the point of today. So here's what we're going to cover in the next 20 minutes or so. By the end of this session, you'll be able to define computational literacy. And yes, it's more than just writing code. You'll be able to articulate why this matters to your students and identify 3 big reasons why it hasn't made it into B&B classrooms yet. You'll understand a framework called TPACK that gives us a useful map for thinking about what we need to know to teach coding to BMB, biochemistry, and molecular biology students. We'll also talk about some specific teaching strategies, notebook-based learning, and live coding that make this whole thing a lot less scary than you might be thinking. And finally, I hope that you'll identify one concrete step that you can take when you get home, not a 5-year plan, but just one step toward working toward coding in your B&B classroom. Should we be teaching coding to life scientists? We actually published an editorial in BAMBED with the title, "The Elephant in the Room, Should We Be Teaching Coding to Basic Science Students?" And I like that title because it's a great question, and nobody wanted to be the first one to ask it out loud. So my colleagues and I did. We asked that question out loud, and we surveyed B&B faculty, people just like you and me, and found that more than 95% of them said yes, it's important for their students to learn coding. 95%. But then we asked another question. Have you ever actually run a coding activity in your classroom? And 62% said no, never. So we have this enormous gap between what people believe is important and what people do with their students. And that gap is why this workshop exists. And so the good news is that you're here, right? You're going to be part of narrowing that gap. Excuse me. There we go. It's more than just writing code. So defining some terms, computational literacy gets thrown around a lot, and it can mean different things to different people. We use a definition from Andrea de Sessa, who argues that a new literacy emerges when a broad cultural group adopts a new tool or new way of reasoning. And given how computation has exploded across every corner of science, we believe we are in the middle of an emergent computational literacy right now. I also like that computational literacy has 3 components. It's not just the material part, being able to read and write code. There's also the cognitive part, using computation to analyze and solve problems. And there's the social part, communicating through computation. Commenting your code so a collaborator can follow it, sharing a notebook with a student, documenting what you did. If you've ever written a methods section for a paper, you've already understand the social component. It's the same idea, it's just applied to code. So why now? Why here in 2026 when computation has been a part of science for decades? Computation in STEM is not new. There are papers about it in the Journal of Chemical Education in the 1980s. So what's different now? What's different now is the scale. AlphaFold changed structural biology essentially overnight. Next generation sequencing went from this exotic tool to a standard method in many research labs. The amount of data being generated in fields like bioinformatics, computational biology, and protein design is simply too large to analyze with Excel. Although we use, although we often use Excel for a variety of problems, it has its limits. There's also broad support for building computational literacy. The American Society for Biochemistry and Molecular Biology explicitly recognizes computational skills as a necessary part of BMB curricula. And the Department of Labor has developed training to foster an AI-ready workforce. Computational literacy is not an extreme idea, it's rapidly becoming the standard. The students sitting in your classes right now are going to work in a world that looks very different from the one we trained in. And we have an opportunity, and I would say a responsibility, to give them the tools that they need to succeed. We surveyed BMB faculty about what topics they'd most like to introduce coding around their courses, and the results are shown here. 40% said plotting in Python, 37% said molecular visualization, and another 30% said working with Google Colab. What this says is that the community of educators is asking for fundamentals, and that's exactly what we're doing today. So I think this is exciting. And for our complete beginners, I hope this is reassuring. It's fundamentals. Why hasn't coding made it into most BMB classrooms? All right, so 95% of faculty think this is important. Why aren't we doing this already? Well, we've identified 3 reasons. First, coding is perceived as complex. And yes, it is complex, but perceived complexity and actual complexity are 2 very different things. I'll speak about this next. Second, differential access to computational resources. Not every student has a laptop, not every lab has good computers, and not every institution has the same infrastructure. There is a real equity issue here, and that demands a real solution. Third, faculty are reluctant to teach what they don't know. And this is completely rational. If I walked into your lab and asked you to teach a technique you'd never done, you'd look at me like I had 3 heads, and the same principle applies here. So all 3 of these barriers are real, and all 3 of them have answers. So let's take a closer look at each one. I don't even know where to start. This is the one I most often hear from beginners. And we understand this. But again, the perceived complexity is often much higher than the actual complexity of getting started, especially now. Because the environment we're using today, Google Colab, completely changes the starting line. Colab lets us give you notebooks with some or all of the code already there. It allows you to run that code in small sections with clear instructions on how to make simple modifications. Think about it this way. You don't need to know how to build a car to drive one. You need to know enough to get where you're going and enough to recognize when something's wrong. That's what we're building today. And look at this example on the right side of the slide. This is actual code from one of our notebooks. Even if you've never written a line of Python in your life, you may even be able to read it and get a sense of what it's doing. The font may be a little small, and I apologize. But readable notebooks are not an accident. Readable notebooks are part of the design. Not everyone has the same resources. This point is really important. Computer access at higher education institutions varies enormously. Some students have brand new MacBooks. Some students are borrowing a laptop from the library. Some school computer labs are running Windows. Some are Mac. Some students have Chromebooks. And some computers, a lot of them, don't have the processing power to run code without freezing up, which is obviously, I think, detrimental to student learning. We've chosen, I think, an elegant solution, Google Colab. Colab runs on Google servers, not on your computer. So it doesn't matter if you're on a 5-year-old Chromebook or a brand new Mac Pro. All you need is a browser and internet access. The practical implication of that is quite substantial. Any student at any institution can run the exact same code on the exact same data as any other student. This technology genuinely levels the playing field, and that matters a lot for student learning. I can't teach what I don't know. That's kind of the big one. This is, again, very rational. Carbona and colleagues identified this as a primary barrier to classroom change. Van Wingarden and colleagues found that lack of time, training, and professional development were the top concerns for faculty trying to implement new teaching practices. This may all sound familiar. It certainly does to me. But I hope you'll hear this and take it to heart. Nobody here is asking you to become a computer scientist. Nobody is asking you to walk into your biochemistry course next fall and teach a semester-long coding module. What we are asking, and what this workshop is designed to give you, is enough foundation for you to take one step. Try one activity. Show your students that coding is something that their BMB professor does, and therefore is something that they can do too. The notebooks we've built are designed so that you can run them without knowing everything that's in them. You can learn alongside your students, and in some ways that can be a fantastic pedagogical model. What do you actually need to know? I'm going to introduce the TPACK framework here. It stands for Technological Pedagogical Content Knowledge. TPACK was developed to help teachers figure out what they actually need to know in order to teach effectively with technology. And the key insight that motivated its development is that most educators use technology to improve efficiency. For example, PowerPoint and learning management systems like Canvas or Blackboard, rather than to augment deep learning. TPACK asks us to think more carefully about that. There are 3 domains: content knowledge, that's the BMB, biochemistry and molecular biology expertise that you have. Pedagogical knowledge, that's that you know how to teach, whether that's lectures or active learning. And then there's the technical knowledge, TK, and that's your knowledge of the tools. Finally, there are the intersections. The one we care most about today is TCK, technological and content knowledge, which is using tools like Colab and Python specifically in the context of BMB. Because knowing Python in this abstract is very different from knowing how to use Python to analyze a Bradford essay. So the goal today is to give you this TCK, but we'll also start building some TPK, the knowledge of how to use tools to help your students learn. Be on the lookout for future workshops that will focus more on TPK and TPACK at the intersection of all of the zones for BMB teaching. So how does all of this map onto what we're actually doing today? First, we will become familiar with GitHub and Colab. That's TK, pure technological knowledge, getting you comfortable with the tools. Python for BMB data analysis, that's TCK. We're not learning Python in this abstract environment. We're learning Python by fitting a standard curve, by calculating Michaelis-Menten kinetics, by analyzing plant growth experimental data. All of these concepts are grounded in BMB. The notebook on linear regression is also TCK and introducing TPK, because you're not just running the code, you're thinking about how you might explain this to a student. By the end of today, you will have explored a little in all 3 zones of TPACK and several intersections. Not perfectly, we can't do it in one afternoon, but remember, we're just taking that first step. Understanding the pedagogy behind a workshop may help you think about how to use similar approaches with your own students. There are 4 principles driving everything that we're doing today. First is context first. Every concept we teach is grounded in BMB. Variables and loops, we can use them to calculate Michaelis-Menten velocities. Data frames, we're going to use them to organize experimental data. Hopefully, every block of code is relatable to a BMB context. Second, incremental complexity. Starting simple and building on that. We don't want anybody to feel they're left behind, so we're not going to sprint. Instead, we're going to start at a walk and move maybe up to a light jog. Third is live coding. When we write code in front of you, we are going to make some mistakes. And that's actually on purpose. Well, it's also unavoidable. But watching someone debug in real time is one of the most educational things a beginner can see. It normalizes that process. Even experienced coders get error messages, and they get a lot of them. Fourth, you'll leave with something real. Besides a certificate, you'll have access to a series of working notebooks that you can use in your own research labs and maybe modify for your own B&B classes. So I'll close this presentation with this. By the end of this afternoon, you'll have explored Colab and GitHub. You'll have developed some foundational Python skills for what we do in B&B. You'll have notebooks that you can hand to a student next semester if you want to. And you'll be part of a growing community of B&B faculty who are on the same journey. I'll end with this quote from our grant. Developing computational literacy in B&B students is important for the next generation of scientists to fully participate in evolving scientific fields. I think that's true, and you being here today, learning something new and willing to be the beginner, that learner, it demonstrates your willingness to grow. You don't need to become a software engineer. You just need to be confident enough to take that first step, which is exactly what we're doing today. So I'm going to switch over to my next talk, which will be a combination of a walkthrough and a PowerPoint. It'll be both parts of GitHub, but we've got a few minutes and I want to be able to adhere to the schedule so we can answer any questions at this time also.
And at this point, your microphones are not turned on, but you can put your questions in the Q&A. And I went I went through the list of attendees, and I recognized at least 15 of the usual suspects, people that I've known for many years. Wally and Chris know we know each other, and so yeah, I'm sure you have questions, curiosities, concerns. Even talk about a barrier that seems daunting to you or whatever. So please have us some questions. We've got about 5 minutes for questions here.
I see, sorry, I'm reading from the bottom up. Sorry, let me go back to my Zoom. Do we need to install Python? No, that is another great thing because we're going to use Python on Google Colab servers. So we do not need to install Python. It's one of the great things.
There's a question here about how private or secure are Google Colab notebooks? Since they run on Google servers, are they accessible by Google engineers? Can they be used to refine Gemini? I can't answer that. Chris, Wally, do you have any feeling for that?
That data is on Google servers, and so they, in theory, do have access to it, and it could be used in Gemini.
Okay. Another question. For those who have run this, what do the students find most challenging? In my lab, it's just getting started, because they think they have to get the right answer right away, and another challenge is they're used to using Excel and Word, which do exactly what you tell them to do when you tell them to do it. And they typically don't see error messages. And when they start doing something in Python in a Colab, things don't work sometimes, and they have to wrestle with that. So the idea that just because it's on a computer, it doesn't work the first time you click on it. You've got to learn how to do it. That's what I've seen.
To me, the issue is really getting them to slow down and read the instructions. It's kind of the same problem that they would have carrying out a regular laboratory, which is that they skim it, they jump to what they think is the next step, and when you've missed a step, things don't seem to work right. Encouraging AI coding help. I've experimented with that this semester with mixed results. I think in some ways it allows them to do a lot more. The problem is they don't always understand exactly what they're doing. And they're not creating, I would say, a workflow that's reusable. For example, like when you sit down and they have to solve this, I guess I'll just, to keep it short, I'll say that I encourage them to work through everything on paper first, and I want to see it on paper before they use AI to arrive at that same answer. And then they can use it to calculate multiple things. But there's a lot more there.
And I would say the
Wally,
AI only is accurate maybe 80% of the time. And so students will more often than not get it wrong if they're using AI.
got another question here. How do we practice with big data sets with Colab considering no high performance computing infrastructure? Chris, can you answer that?
I can. There is a whole website of big data sets that you can download and work with for free. I'll try to get that and give that out to the chat so you all can take a look at it.
And I suppose some very strong detailed computations are not going to be able to be carried out in this environment without a high performance cluster. However, there's a lot you can do, right? You can submit individual protein structures to AlphaFold and things like that. So a lot of big data you can actually process in this way.
And Chris, you showed us in Colab, you can actually select a different processor to get some more juice, right?
Yeah, you can change the processor, and I'll show everyone how to do that in a couple minutes when I do my collab.
Okay, another question. How can I join the BMB community? I'm assuming you mean the PyBMB, so the coding community that's working in BMB. You can email us at codingbmb@gmail.com. And when you do that, we will invite you to join our Slack community, and we'll invite you to join LinkedIn. We have a LinkedIn community as well, and so we can get you going with us. Wally, you want to handle the next one?
Much Python is required for a person who knows essays. Okay, so for our notebooks that we're going to cover today, we're going to utilize Python. But you can do a lot of wonderful exercises. And actually, I think Chris should talk about this because he does a lot of exercises in R. Chris, do you want to reflect on some of that?
Yeah, I came from R originally. I learned that as my first language, and then I picked up Python to do the things that R couldn't and to do some more other tasks. And if you know R, Python is not a big leap. And Colab also runs in R, and I can show how to set that up when I get to Colab a little later on.
All right, we've got time for one or two more questions. A question here, do you think Google Colab is enough, or do we also need to learn some Linux tools? Google Colab is a good place to start. And you can get comfortable with Python or R coding in Google Colab. If you want to do more high-powered stuff, you're going to need a more intense environment. Would you agree with that, Wally?
I would say I have almost completely stopped using Unix and my own installations of Python with the advent of Colab. But I would say what Paul is saying is correct. If you wanted to run some code that's going to run for several days on a local computer, you'll want to learn how to install your own Python and some Unix. But you can do so much with Colab right now.
Last question we're gonna take for right now is: are there plans for this workshop to eventually touch on Rosetta for protein design and working with XML scripts? That's not part of this workshop today or the workshops we have coming up in May and July. But if there's demand from the community, we'll develop materials about this. And that's my long way of saying I really don't know anything about XML scripts or Rosetta for protein design. So we'd have to go out and learn how to do that ourselves before we could offer a workshop on it, but I'm sure it could be done. At this point, I'm gonna mute myself and disappear and turn it back over to Wally to continue his next presentation.
I will do a quick follow-up to that. Oh, and now I'm blanking on it. So there's it's PyRosetta. PyRosetta exists, and I think it's Brian Kuhlman has actually put a lot of notebooks up there that kind of run in Colab. Not everything that they have done in Colab, had done works, but there is a lot that you can do already with PyRosetta. Yeah. All right, we're turning now to GitHub. So I'll be honest with you. When I first heard about GitHub, I was afraid to use it. It sounded like something that you needed a degree in software engineering to use. Didn't sound like it was for biochemists. I thought I would be messing up somebody else's code if I used it, or I was going to ruin my own code. But GitHub is not just for curating software. And in the next 30 minutes, I'll show you why. And the good news is most of what you need to do in GitHub today requires no account, no setup, no prior knowledge. If you can use Google, you can do almost everything we're going to do today. But getting comfortable with this platform is worthwhile. And so we're going to demystify GitHub. Here's where we're headed. By the end of this session, you'll be able to navigate a GitHub repository, meaning you can just find the stuff that's there. You'll be able to open any Colab notebook that's public from GitHub with or without an Open in Colab button. You'll know how to build your own Open in Colab button, which sounds fancier than it is. And if you have a GitHub account, you will be able to fork a repository and save your work back to it. That last part, forking, does require a GitHub account. I'll tell you when we get there. If you don't yet have one, you can watch along and set up one later. It does only take a few minutes. Okay, let's start with some basics. What is GitHub? I think the easiest way to think about it is a combination of things that we already know. It's like a shared drive, like Dropbox or Google Drive, except for it's designed really specifically for code and text files. It's also like a lab notebook because a single change that you make is recorded. Who made it, when they made it, what they changed. You can go back in time to any previous version of your code, which if you've ever accidentally deleted something important, is really helpful. And it's kind of like a publishing platform because you can share your repositories publicly and anyone in the world can access them, use them, or build on them. One reason we use GitHub is that it integrates beautifully with Google Colab. You can open a notebook directly from GitHub into Colab with one click, and that's going to change how you share your teaching materials. Before we start clicking around, I want to make sure we're all speaking the same language. There are 5 terms that come up constantly when people talk about GitHub, and I want you to know what they mean. The first is repository or a repo. It's basically your project folder. It contains all the files related to a project, code, notebooks, data, or documentation. Everything is just there in one place. The README is the front door of that repository. It's a file that will appear on the main page of your GitHub page, and it tells you what the project is and how to use it. You should always read the README first, and it can help you just save a lot of confusion. A commit. A commit is a saved snapshot of your files at a specific moment in time. And there's a short message usually attached with it describing what you changed. Think of it basically like when you save your document, it also has a sticky note attached to it that tells you what you changed. A fork is your own personal copy of somebody else's repository. You can do whatever you want in that fork and you'll never affect the original. It's a very important concept. We're going to use it. Now, it's not that you can't share it back with the original, but there are a lot of steps involved. It's something that you can just play around with and make it your own. Finally, there's this thing called a branch. And for now, you just need to understand that branches exist and that the one that's called main is the one that you're almost always going to use. You don't really need to think about branches, but it's effectively just a parallel set of a parallel notebook or a parallel repository. A parallel branch of that repository. Okay, here's an anatomy of a GitHub repository. And this slide shows you what it would look like. It's what it's going to look like because we're going to go there in a minute when you land on it. At the top, you see the repository name, April 26th, Rutgers RCSB. Below that, you see a list of the files and folders. So it's just like a file browser. Folders are first and then the individual files. Our repository has a folder for images, a folder for data files, and then it's got some presentations and 4 notebook files. The notebooks end in .ipynb. And these are the files we'll be working in. Finally, there's a readme.md at the bottom. And that readme contents, they don't appear on this page right now. But when we go to GitHub, you'll see that it automatically renders and displays that on the main page to make sure that it's always your first stop. In the next few minutes, we're going to open this exact repository together. So keep this image in mind. It'll look familiar when you get there. Oops. Okay, here's our first walkthrough. You'll notice that orange badge in that top corner. So that's anytime you see that, I'm going to do a walkthrough live. But I'll explain to you what I'm going to do and leave this up, and then we'll walk through it together. So the first thing we're going to do is open a browser. It doesn't matter which one, but as Paul said, Chrome is kind of native to Colab. But if you have Chrome, Firefox, Safari, it doesn't matter. You're just going to go to github.com. When I do this, I'm going to do it in an incognito browser, so it won't have me logged in. In the search bar at the top, we're going to type CodeBMB and press enter, and then we're going to see a list of repositories that user, CodeBMB, has. And we're going to click on the repository called April 26th, Rutgers-RCSB. Once we're in there, we're going to take a minute to look around. We're going to scroll down. We're going to read the README. We're going to find a file called 001 Intro to Google Colab, and we're going to click on it. And that is going to give us a preview of what that notebook looks like. It will look a little bit different than Colab, but it'll be very recognizable. I want to emphasize one thing specifically. When we do this, everything that we're going to be doing, looking at all this code, we're doing it without signing in. We didn't create an account, and we didn't install anything. So we can just do all this through a web browser. All right, let's go ahead and do that. So I'm going to try and switch here. So we should see my incognito window now. And I've got my instructions over here. So we're just going to go to GitHub.
We're still seeing the walkthrough slide, Wally.
Oh, is that right? Okay, so I can change this. So that's still very easy. I appreciate you telling me. I thought that was too good to be true. Now you should see my incognito window. Is that true?
Yes, it is, Wally.
All right. So I'm just going to go to github.com. And this is, if you don't have an account, this is what you'll see. But there is this search bar here. So I'm going to click in it. And I said we're going to just search for code BMB and hit return. And we get one result. And I'm going to click on this link. And so you can see that this looks very similar to what we were showing before. It says I'm in CodeBMB, and it says which repository of CodeBMB. This is everything I showed you before, but I can click on them. And here's the README file down at the bottom. Automatically displayed for us. So what we're going to do now is click on Intro to Google Colab. And that's going to preview what this would look like in Colab. It does have this Open in Colab button. We are not going to click it yet. But this preview shows us what this notebook looks like. And we can read through it. The code, I'm switching up here. The code view will look very, very similar to the preview. I'm going to click on the blame view. Blame is just what it sounds like. If you want to know who broke your code, it will tell you. The code here is actually written in this markdown in this coding language so that it can display it as a notebook. But you can see that Chris Berndsen made a lot of changes 28 days ago, and that's exactly what he did. And so we can see all of the changes that he made. That's just to show you what that is, but the preview view is what we're interested in today. Okay, I'm going to jump back into Zoom. Oh, sorry, into my PowerPoint. Okay. Here we go. Okay, so we're going to do a second walkthrough, which is actually opening that notebook. And I'll do this first version here, option A, which is the easy one. A lot of notebooks have this open in Colab button right at the top. So if you see that, you can click that and done. It's going to actually, as long as you're logged into your Google account, it'll open it right in Google Colab. Otherwise, it may ask you some questions like to log in or to open them. So this notebook will open directly in Colab and you're ready to go. After we do that, you'll go to File, Save a Copy, and you can save a copy in your Google Drive so that you have your own editable version. Let's go ahead and do that. So I'm sorry I'm jumping back and forth so much, but I think it'll be helpful. Find the right. All right. So I'm back in my Google Chrome. Actually, I think I'm gonna stop this again because I want to share a different window. Okay. The reason I'm going to share in this window is because now I'm logged into my Google account, and so things will just log in for me automatically. So I'm going to go ahead and navigate back to that repository. So if I go to github.com CodeBMB, I can just click on the repositories button, and I see now April 26th. All right, so we're back in this repository. We clicked on the intro to Google Colab. And in this preview view, there's this open in Colab button. And I'm going to go ahead and click that. Boom, it opens. And now this is actually ready to be run. And what I said is we're going to do File. So here inside the browser, I can say File, save a copy in Drive. And that's going to open up a new window. And it's now called Copy Of. But this is your own personal copy of this, and it is saved in your Google Drive. Okay. All right, jumping back here. Option B, if there is no button, and we'll do this together, if there's no open in Colab button, it's still incredibly easy to open the lab. We're going to just type in this address in our browser, colab.research.google.com, and then we're going to do a file open notebook. Sometimes it opens with a window that's asking you to open a notebook already. We're going to click on the GitHub tab, and then I'll show you a couple of ways. You can either put in the entire URL, or we can just search for CodeBMB. Either way works just fine. So let's go ahead and do that one now, where we're going to pretend there was no open in Colab button. All right, I'm back here in my Chrome window, so I'm going to open up a new window, and I'm going to type colab.research.google.com. And I'm going to hit return. And it kind of gives me this generic background. And then I have some examples of where to open a notebook. I can upload one from my computer. I can get it from Google Drive, or I can get it from GitHub. Now, currently, this is just searching for me. I could paste the entire URL. It's got to have the HTTPS in there, the entire URL. Or I can just type CodeBMB and don't hit enter, but click the little search button. And here we go. It found, I can select different repositories there. We're going to do obviously this April 26th Rutgers one, and you can see all the notebooks in here. So I can open it just by clicking on this. So that's another way, and I can do that with any public notebook. Again, I can do File, save a copy in Drive, and make a copy of this. That will be my own copy. Okay, let's get back to the presentation. Okay, this is just highlighting how easy this is to do with any GitHub notebook. Just first going back to that colab.research.google.com, and you can either do File, Open Notebook. Again, if you may not need to do the File Open if it pops up that screen initially, but you're going to click on that GitHub tab, and you're going to either paste the repository or search for that username and press Enter, and that's all you've got to do to open a repository. And that works for any public GitHub notebook. So it's exceptionally helpful. Okay, we're not actually going to do this one today because I want to focus on some other things, but I wanted to share that. That Open in Colab little button, this is what it looks like. That badge is just a line it's called Markdown, so it can go in your notebook. You can add this and it will display as that Open in Colab button. And you just replace username with whatever the GitHub username is, repo with repo, blob just stays blob. I don't know what it means or why it's there. Branch, which will almost always be main, and then whatever notebook it is with the .ipynb, and that will open that notebook automatically in Colab. So it's really easy to kind of add that shortcut. I suggest adding it to all of your notebooks because it's really easy for students and for you to access. But remember, if you don't have it, it's very easy just to do File, Open, and search for that repo and file. Okay, you don't need a GitHub account, not for everything today, but yes, you would need one if you want to be able to share things here. So this is just to be clear about what you can do with and without an account, because I don't want you to feel like you're locked out of something. But without an account, you can browse any public repository, you can read any file, read any public notebook, and download any public file. Everything we've done in these last walkthroughs, there's no account required. So a student to run a notebook would not need any account. But with an account, you can fork or copy that repository. You can save your own work from Colab right back to GitHub. You can create your own repositories, and you can share your own notebooks publicly. Creating a GitHub account is free and takes just a few minutes at github.com. So if you haven't done it yet and you want to do the next walkthrough with us, now is a great time to set that up. Otherwise, if you don't have a GitHub account and you want to wait, you won't be lost without it. There's going to be a video, but you can watch along and follow up later. There'll also be these PowerPoints shared with you. But the next walkthrough is kind of useful for teaching. So there's my warning. It requires a GitHub account. So if you don't have one yet, just watch along. It'll take it'll take a few minutes. So, but first, what is a fork, really? A fork is your own personal copy of a repository. It lives in your own GitHub account. You can edit it, add to it, personalize it in whatever way you see fit, save your own notebooks, and none of that will affect the original CodeBMB repository, which is really great. You can do with it whatever you want. And so here are the instructions, and then we're going to go through it together. You'll go to github.com, CodeBMB, April 26th, and then you're going to look for the fork button. It's kind of in the top right corner, and you're going to click that. You'll leave all this default settings alone and click Create Fork. GitHub will redirect you to your own copy of the repository. The URL will now say github.com/users/fork. your username, April 26th, Rutgers RCSB, and that'll be your copy. Now, when you go to Colab and open that notebook from GitHub, your forked repository will appear in the list. You open that notebook, you can work on it and modify it, and when you're done, you'll go to File, Save, and you'll save your copy back to your fork. That's the workflow. Fork it, open it, work on it, and save it. Okay, so we'll break out, and I'm going to walk through this again. So I'm going to close some of these windows, and I'm going to go to github.com CodeBMB. I'm going to go to this repository that we've been sitting in, and I'll just wait for just a second. Well, 23 of us have forked it already, so there we go. Here's that fork button. So it tells you how many people have forked this repository, and all you have to do, if you've got a login, and if you don't right now, again, that's okay. But we'll just see what this looks like. We're going to click that fork button. And this says it's going to give a fork under my username, right? And then it's going to give it the same repository name. And I'm only copying the main branch, and I'll click Create Fork. So now GitHub has in fact redirected me. If we look at the top of my screen, it says I'm in github.com Wally Novak, not CodeBMB, and Rutgers RCSB. Wally Novak, April 26th. And notice this one has not been forked at all because this is my copy now. This is not the CodeBMB copy. Okay, I do want to hit on there was a last little warning there. If I go into this, into my forked version, this intro to Google Colab, and if I try to click on this open in Colab button, that actually still points to the CodeBMB. And if you look, I'm going to move my mouse, but if you look down here at the bottom of the screen, when I put my mouse over it, you'll see that that's actually pointing to the CodeBMB version. So we don't want to push on that immediately. We want to use that walkthrough to method, which was, I'm going to go to Colab, research.google.com, and I'm going to go to GitHub, and I'm going to go to Wally Novak, searching for my own repository, and you see April 26th, and now here's my version of Notebook 001. If I open that here, you'll notice that I don't see that open in Colab button. It's because it's added automatically by Google. So when I go now and say File, Save, just File, Save, it's going to ask me, okay, save it here, Wally Novak. It's saving it over that file. This is where this changed the open in Colab button to work for my fork. And I've checked include a link to Colab. So when I click OK here, and I go back, you'll now notice if I put my mouse over that down at the bottom, it says Wally Novak. This is now ready to go. It's my own version with my own open in Colab button. And just to show you, I guess, I'll go ahead and click it and open it. You can't tell where it is other than up here, github.com Wally Novak. So now that button works for my version. Okay, in the last few minutes, that's my last walkthrough. But in the last few minutes, I need to share some information about, saving files. And we went over one already, so that's good. Lost my instructions. Here we go. So there are a few options to save your work. That first is Google Drive. File, save a copy in Drive. This is the easiest one for everyone to do today, especially if you don't have a GitHub account. Your notebook saves to your Colab notebooks folder. If you don't have one, it will make one in Google Drive. And then you're done. You've got it. Option 2 is just file save. It will go to your GitHub account and it will save it directly to your forked repository. It also creates that commit, which allows you to, again, save a little sticky note about what you did. It's that time-stamped snapshot of your work. Option 3, download it. You can download it directly to your computer. The problem with doing this is a great backup, but the problem with doing this a lot is it creates an extra friction step because you always have to go upload whenever you want to use that again in Google Colab. So again, my recommendation is to use Google Drive. It's fast, it's simple, and keeps your work safe. If you have a GitHub account and want to use option 2, go for it. But don't let that GitHub workflow slow you down during workshop time. GitHub is a scientific tool. You can think about this as a notebook for your code. Every analysis that you do, every modification you make is tracked and time-stamped. If a reviewer asks you how you got a particular result, you can show them that exact code, the exact data, and the exact version. So that helps a lot with reproducibility. It's also a tool for sharing with your students. When you post a repository, you can either have your students fork it, right? And they can work on their own copies and share their results with you, or they can click the Open in Colab button and share their results with you that way. There are no I can't open your file messages and no version confusion. It's also an open science platform. More and more journals are asking authors to share their analysis code alongside their papers, and GitHub is how a lot of scientists can do just that. And finally, it's about a community. So the CodeBMB notebooks, everything that we're working on, they live on GitHub. When we update them, you can get those updates. It'll actually let you know that there's an update in your fork, and you can import those to update your own code. If you generate some improvement for us, you can tell us, and we can import that code and update our version. And that's the whole point of GitHub, being a community of coders. Okay, that brings me to the end. I think right on time. A quick inventory is, I hope you can navigate to any public GitHub repository with no account needed. Open any notebook that's public in Colab with or without the Open in Colab button. There's some instructions for how to build your own Colab badge. But remember, if I'm in Colab, I can just check the box, include Open in Colab button. And if you have a GitHub account, you can hopefully fork the CodeBMB repository and save your work back to your own copy of it. Remember the 2 things on light blue, they're the ones that you need an account for. If you haven't set one up yet, do it this week or on the next break, which will be after Chris's section. Again, it takes just a few minutes. And remember where our repository is. That's at the CodeBMB in GitHub. So bookmark that. All right. I think with that, I'm going to turn things over to my colleague, Chris Berndsen.
We're going to start working with these various collabs. There are 4 different sessions that we will be learning, and we're going to build. This first one here is going to talk specifically about Colab, what it is, what it isn't, what we can do with it, and then set you up to start working with some data in a session on basic Python coding, and the one I'll do later on Pandas, and then one where we're looking on data fitting later on, or the last one done by Wally. So we'll be working with Colab the entire time. And as a reminder, once you open up the notebook, go ahead and save a copy and drive. I've already got mine split out, but there's a save and copy and drive right here. Click on that, and that way you have your own copy to keep. If you don't do this, when you close out of the Colab, you will not be able to come back to this and use this any further. So go ahead and just save a copy and drive right now. Now what I have here is Google Colab number 1. It's in Chrome, and we've got all these different things showing up. So let me orient you a little bit, and then we'll start working our way downwards. Across the top is our menu bar. You've got File, Edit, View, the standard sort of options along with Runtime and Tools. We'll look at these two in just a moment. We have a Share button, just like you've seen in Google Docs before at the top right, along with a Connect button, which we'll explore a little later on, and then a Table of Contents here on the left. Now, the Table of Contents isn't visible for you. There's a menu bar that has 7 buttons on the left side here. For me, if you click on the Table of Contents, then it'll come up, and you can use this to jump to different sections. This is great, especially if you just want to come back and look at one section later on. There are other sections here that we'll talk about the files in just a moment, but we've got Table of Contents here. Now, what I'm going to do is hide the Table of Contents just so my screen's a little bigger, and I'm going to make the text a little larger as well. That way it's easier to follow along. So this is Google Colab. I use this for teaching. I've used Google Colabs in my classes for about 5 years now, and I use them to do basic calculations in Gen Chem types of settings, but I also do them for project-based learning and cures in biochemistry lecture and lab. And I'm using this on a scale of 20 to 120 students usually. So especially my lecture project, where I've got well over 100 students, we're using Colab to do basic calculations, but also running structure prediction, molecular dynamics, docking, some really sort of advanced applications. We're not going to get to those today, but we will be sort of heading in that direction of we want to learn how to do the fundamentals so that in future workshops or maybe even on your own, you want to explore this. So at the top, we've got our header piece that explains, gives you some of the details of the Colab. And this is really good to have in general, to have an explanation of Colab and what it is. And section 1 talks about how to make a copy. Already had you do this, but if you want to do this again, you go to File, Save a Copy in Drive, and that'll be a good way to get you started. Now you can rename it by going up here to the top bar and just changing the name to whatever it is. So we'll call this Live, because that's the one I'm using right now. But just like a Google Doc, you can change it right up here in the top. Now, like a Google Doc, Colab autosaves every couple of minutes, and it saves it to the cloud and saves it to usually a folder in your Google Drive called Google Colabs or Colabs. It happens in the background, but if you just did something and want to save it, say you know your Wi-Fi is going to close out or you want to close your computer, you can do a manual save, or you can update your copy or click save down here. Either way you want to do it to make sure that you have that. I have found sometimes, especially if you're using Wi-Fi and it's not really strong Wi-Fi, that saving can happen somewhat irregularly. So it's always a good idea to make sure that you are saving it before you close your computer for the day. Another alternative is something that Wally mentioned where you can download, and that's under File, then Download, and then you can save it as an IPYNB file, and then you could upload it back to Google Colab if you needed to, along with being able to save it to GitHub. But you've got a lot of options for saving your work here. Use them as you feel appropriate. Now, Colab is broken into a couple of different types of what are called blocks. And on my screen, I have section 3, and then there's this blue border going around it. This is a type of block. This is an example of a text block, which is written in a type of code called Markdown. And you can stylize your code a lot of different ways. And then there are code blocks. We're going to explore text blocks first. I'm going to double click. Actually, let me show you the better way to do this. So I'm going to create a text box. If I move to the bottom of the block I'm at, and then I move over and click text, it will create a text box that is already open for me. I can then type into it and say, this is a text box. This is regular text. In a moment, it'll render to the right side of the box. So on the left side, you're typing your markdown. On the right side, you are creating text. And there's a style menu across the top here. You can explore different styles. Say I want to change regular text to bold. So I'll highlight bold. And I can do just like I do in a Word doc, or I can type that out and say, this is bolded text, and then go back and add in asterisk manually on the other side, and it will create the bolded text. You can incorporate links and images as well. And if you have questions about how to do a lot of the stylizing, the very end of the Colab, I have in Appendix 1 stylizing text, and it goes through this in great detail and shows you all the possibilities for creating text. And this is a good way to create text that is attractive for others and make it easier to read for students as well. And I'll give you an example of how I've structured a Colab with stylized text and all of that in just a moment. There are also code cells, and these are the ones that run code. If you come down to the bottom of a block and click on code, it will open up a code block, and we can type our first code if you want. We'll keep it very simple. We'll do the standard print, which tells the computer you want it to print something. We'll put quotes in there, and we can type hello world. And then there's a play button here that you can push. Now, I haven't told you about the play button or how to run code, so if you haven't gotten there yet, that's fine. We'll address this again in just a moment. But if you want to try running code in a code block, this is one way to do it. Type print hello world in quotes, and then push the play button, and then within a moment or so, it hopefully will print out hello world. Again, we will address more about the code aspects in just a block or so. Give everyone a moment to catch up or to play around a little bit, and then we'll talk about stylizing or changing the look of the Colab. All right. Now you can change the look of your Colab to be more personalized. And in some cases, you may have already seen that. If you've been opening up your code block, you may notice that you do not have numbers on the left side of your particular Colab. That is because I've already changed the settings to provide line numbers in my code blocks. And so you can do this yourself by going to the Tools menu. Now in the Tools menu, when you click on it, there will be a couple of different options for you. Settings is a good place to start. And you can go to the second option here, which says Editor, and then change what you want it to look like. This includes changing the color scheme if you want, to change the font size, the type of font, indentation. The ones that I like to turn on are line numbers. And this is great when you're teaching, especially if you have a line number and you're live coding for the students, and you can say, go to line 3 of the second code block. That will help them locate it. So I use line numbers and also indentation guides. Students are often challenged when we start adding indents into our code, and Paul will show you examples of that in just a moment. And I always like to keep track of them using the guides. And so you can click these buttons to make them. There's also fun ways to personalize it. You can enable things like having dogs or cats or crabs walk across the top of your screen. Also a good way to keep students engaged if you want. I'm going to leave those off for now, though, because it is a bit distracting sometimes. So you can explore these editing options and also look at some of the miscellaneous options as you want. But again, I recommend going into the menu and turning on line numbers and indentation guides to help you as we're moving forward, as those are key aspects to working through code and reading code and understanding them. All right. Now, in section 5, we're going to talk about the runtime type. Now, we haven't said what a runtime is. A runtime is a computing environment, and you can choose hardware and coding language. So to choose my runtime, I'm going to come over here to the right side, and on my screen it says RAM and disk, and there's a green check mark next to it because I've been running code. You may have connect or something else. I'm going to push the down arrow that is next to this, and I can click on change runtime type. And you've got a lot of options here. Don't get overwhelmed. There are guides. You can click on the question mark and it'll take you to a new screen to explain things for you if you have questions. In terms of runtime language, Python is an option. There also is R, which was asked about in an earlier session, and Julia, which is a different language. Python is what we'll be discussing today, but there also are some great collabs that use R. And then you can choose what type of hardware you're using. We're using a CPU right now because that is all we need for the types of work we'll be doing, not only in this collab, but also in the later collabs. If you were doing something that is more computationally intensive, say molecular dynamics, or you want to run AlphaFold and do model inference or something like that, you would need to use a GPU for that. The standard free GPU is a T4 GPU, and you can get access to some of these others if you are on the paid plan. But for most of what we need to do in biochemistry and molecular biology, the CPU runtime is fine. For some more advanced applications, then you might want to have a GPU. Now, there are limits to the runtime and how long you can be running a process on Colab. On the free plan, it's usually around 6 hours or so, and then it resets daily. But it's I have not had students run into any of those runtime issues or having it time out, meaning that you've exceeded the time, except if they just stayed connected and forgot about it. And then you can do some other options here. But the main ones that you would be concerned with early on are the language and your type of hardware. Once you've made your selections, you can click save and get out of it. You can also look at how much time you might have left by clicking on view resources, and it tells you how many compute units you have available and how fast you're using them and how many sessions you have. This can be important as Google limits the number of active sessions you can have, but also how long or how much you can use per month. I have a Google Colab Pro account because you can get those sometimes as educators for free. So you may, if you are interested in doing this for some more advanced applications, try to get one of those free accounts. So we're going to go with the defaults that this Colab will go to. We won't worry about changing the runtime to run anything from this point forward because the CPU runtime is fine. So now let's talk about the play button. We addressed it a little bit earlier when I ran that test code to show you how a code block works, but now we're actually going to get into the details. Beside each code block is a play button that when you press it, it will run the code. Now, if you ran the code earlier, the hello world code that I ran, you're already likely connected, and you can confirm that you're connected by looking at the green check and making sure that there is one. If it says connect in the top right of your screen still, when you push play, it'll automatically connect and then it'll run the code. So I'm going to run this code. And I could tell it was running because the play button got a little a dashed line around it. And then it worked because it tells me that it was a green check that it ran the cell successfully. If something fails, you'll get a red X, and I'll show you an example of that later on. And then sometimes it's good to know what order the cells were run in, or sometimes you may have skipped one and you run things out of order. It'll tell you what order you ran it in over here to the left as well. So the key thing is the play button and having it run, and then making sure that you get a green check at the end to indicate that you successfully ran the code. All right. So we've run some code and we got a date and a time, and it gave us a message here. Great. Maybe we don't want to run using the standard built-in Colab packages, or we need to tell Colab what software or code or scripts that we want to use. That is called a library or a package, and we need to install them. There are commands like pip, and that can tell you to install a package. And you can find these packages in GitHub or on other Python library sites. And they'll tell you, here's the command to install this. And you can copy and paste that into Colab. Now, one caveat is if you're running in Colab and you're running something like pip, you need the exclamation point in front of it. Tell Colab you're going to run this as if it was a command line. And so we're going to, in this code block, we're going to install this package called emoji, which is a fun one. It introduces graphics into your text. And then we're going to tell Colab that we're going to import emoji. So we've installed it, but now we want to activate it. So you can think of this like in line 3 where we install it, we're downloading the software to our computer. And then import emoji is like clicking on the icon and saying, I'm going to use this piece of software. Once I've imported it, I can then use that, the functions within that package or the scripts or the code within that package to do different things. If I run this one, it will install emoji and produces this little bar. And then once it's fully installed, it will run the code. In this case, it said Python is awesome. So it took my setup around 6 seconds to do that. I would assume that this code will run in about 20 seconds or less. So hopefully it's working for everyone. So now we haven't dug into a lot of code yet, but let's try writing some really basic code using this emoji package. I've got a link here for possible emoji codes. If you click on it, it will take you to this website, and it gives you a list of emoji codes that you can use, and it shows you what it looks like in the left column, and then the right side column, the wider one here, it gives you what the code is and a lot of more details. The main thing we're going to want is that code that is between the 2 colons. And so we can come back here, and I've already got one line written out for you as an example. It says print, and then it runs the emoji function to say, in quotes, I write code on a desktop computer. And when I push on that, it shows that. So why don't we all take a moment to find an emoji code that we want to try, and go ahead and change the sentence to something and incorporate an emoji into it. It's just something fun to do. So, I like to sit on and then we'll go and find couch. Couch works. It auto-filled for me, but we could also go back and maybe I want to make it beach. So I'm going to copy the emoji code here, go back and paste it in and run it. And it runs the first line of code, line 2, and then it runs line 5, which is the one I just wrote. You're welcome to play with this and explore as you want. The key things here are to have print, then tell it to use the emoji software, and then in parentheses and in quotes, type out your statement and see what you get. So I'll give everyone a moment here while I take a quick drink and let you play with emoji, and then we'll move on to code commenting, which we've already seen, but now we'll explain what's going on. All right. Code commenting, for me, is one of the strongest teaching tools that I can use within Colab, in that it allows me to leave notes within the code for my students to look at. And it also allows my students to write notes and tell what is going on in their particular code. So previously, I've mentioned that we have text blocks and then we have code blocks. And so what commenting is, and what we're referring to here as a comment, allows you to write text in the code block that is not run. And the code that you see in my screen on line 1 and on line 4, where I've got the hashtag or the pound sign or the number sign, depending on your generation, followed by some text in green, that is a comment. And anything that has got the hashtag in front of it is not going to be run by Python. It's going to skip over it and look for the next line of code. So in this block, I've got some unannotated code set up that you can practice commenting on. Now, to do a comment, we're going to add the hashtag, and then you can type whatever you want. So in line 5, I've got some numbers there, and I'm going to say create a list. And that can be a good start. Then in the next one, there's do some math. And early on, this may be how you're commenting your code, is just indicating what is happening in each step. And you can even add comments in line with your code. So this is, I'm going to add a comment after line 10 in my case that says, print this text, and try to understand it. Now, what if you downloaded a really cool repository from GitHub and you're working with it in Colab and you don't know what's going on? Well, a lot of times what I tell my students to do is see if the code runs and try to understand what happened by looking at the answer and then looking back at the code and going back and forth. So it looks like by inference, I've got these numbers 0 and 20 and 37 and 100. Well, that's in line 5, so that's my list. Then I did some math, it looks like, to convert it. And then I did something here. I don't quite know what this means yet, but I could start to work out what is happening in that code. You can even interrupt in the middle of code and put your comments in. Let's say this section is the printing section. And the code will still run fine. So use comments as in a couple of different ways. You can use it to help yourself when you're trying to break down someone else's code, if they haven't commented on it for you, to dig into it and say, this is what I think is going on, and then play with it and see what happens. You can help it by helping your students. Say, you need to put a number in here and create a blank spot for them. And you'll see that in a couple of the later collabs. We've put comments in that say, you need to fill in this number. Or you can use it as notes of how you set certain values or why you set certain values a specific way if you're using this for a scholarly work. Maybe you chose a number and you want to put into the code, I chose this number based on this particular citation. And that can be useful as a reminder, just like you would write down in your science notebook or your lab notebook. Having that written in the code can be a good reminder for just good science, and when you open the Colab in 6 or 7 months and don't remember what you wrote. So, all right. Now there are a couple of ways to bring data in. So far, we've just been typing code directly into the Colab, but maybe we want to analyze data from another location. And Paul will show you a little bit about this, and then I'll show you a little bit more down the road about how we can work with data and import it. So one is we can directly upload our files to Colab. On the left side here, we've got these 7 icons. One of them is a folder. If I click on the folder, I've got a workspace here, and I can drag a file into this space and then access it in the Colab. Another way is you can connect your Colab to Google Drive directly. And this is where most of my students do their work, is we load Google Drive and they've got a Google Drive folder and they connect the Colab to that folder and they're just working with data in Google Drive. This is because many of my students are native to the Google environment and so they understand Drive and how it works. And then the third way is the way that Wally showed you, which you can import it from GitHub. And you can either pull the file in individually, or you can pull in the whole repository or copy the whole repository, clone it, if you will, and then work with the data from that repository. One note, and we mentioned this earlier, but I want to mention again, the data upload to Colab is not private, and Colab is not compliant with privacy laws like FERPA or HIPAA. So don't put data up on Colab that is not anonymous and proper. Don't do any data analysis in Colab with data that contains private information or proprietary information as it can be seen by Google and anyone at Google. So this first section here, we're going to connect to Google Drive. I've got the code written out that will do it down below here. But mounting to Google Drive gives Colab access to personal files and folders, shared drives, and any data sets. But you do have to tell the Colab where to go, where to connect. Now, when you press the Mount Drive, it's going to bring up a warning. It says, connect to Google Drive. Do I want to permit Google Drive to access these files? I'm going to say yes, connect to Google Drive. And then it's going to go through a series of screens that I don't know that you all can see because they're not running in Chrome. It's going to ask me for specific permissions. And then it will mount the drive and it will give me a positive response if I've clicked all the boxes right. So while it's running, just taking a moment here. There we go. We've mounted Google Drive. It says now my files are accessible at Content Drive and My Drive. That shows up here on the left side in your files. And so I could click through this and find where files are and see what happens. Now, I'm going to run this code, which is going to just spit out a list of files that are in my Drive folder, just to give you an example of how it has access. But you can decide to do this or not do this depending on how comfortable you are with giving Colab access to your Google Drive. So I'm going to run this, and it's going to go and says that in my Google Drive, I've got folders entitled these various things, and I've got a lot of folders. If you had files, it would show you files specifically. There's lots of different content that may show up, but I've got these set up in these ways. You can also pull in data directly from GitHub. I'll show you one way. I think Wally's going to show you a slightly different way to do this. But you can try a few of these different ways. One is the way that Wally showed you about opening a notebook directly from GitHub. Another is cloning the repository, which is where you copy it and then give your Colab access to it. Or you can pull in individual files. And that's what I'm going to show you here is we're going to go into the CodeBMB repository for this particular workshop. And we're going to pull in a spreadsheet that's already there and import something called Pandas, which I'll explain more about in a later session, and then we'll display it. And so when I run that, it pulls in the file pretty rapidly and then displays it down here. And I know I'm running short on time, so the last bit I will explain is about sharing your notebook. If you want to use this for a class or sharing it with specific people, you can do the GitHub method, which I highly recommend. The other way is more like a Google Doc, where you click on Share, and then it brings up the permission structures that you have, just like with a Google Doc, where you can change it to anyone with link, and you can choose whether they're a viewer, a commenter, or an editor, and that will set you on your way. So that is the intro to the Colab. The sections we didn't get to are about using a form, exporting your data, and then doing a little bit with some of the AI. Those areas, if we may have time a little later, we might be able to go into, but I don't think we'll have time now to try to keep us on track. So I'll stop here, and then we can figure out what is going on next.
Thanks very much, Chris. That was terrific. So one of the questions that I'm seeing a lot of is, how do I connect with this community? So I've been sharing the links in there quite often, and I'll also put those in the chat window so you can see that. So there's a question here: how can I get into the GitHub? So to get into GitHub, I'm just answering this one live. I'm going to share my screen, and I'm just going to pick Google Chrome. Okay, that one. And so if I just go in here and I just type github.com, then it takes me to GitHub. Now, I'm using GitHub today, so it takes me to my repository. But if I wasn't a GitHub user, it would just take me to the general github.com interface. And so that's how you access that. Okay? Okay, there's a question here. Can you show how to mount a shared Google Drive? Chris, could you do that?
I don't know that I have any shared Google Drives to do it, but what you would need to do is create a shortcut to that shared drive in your own Google Drive. So let me see if I can get Google Drive set up like that. But you effectively have to have that folder in your own Google Drive to make it work, and then you can get through it the same way that I talked about, where you would provide the file path for that. Let me see what I've got here. I've got a shared folder here. And if I come all the way over to the far right side, the triple dots, I'm going to click on that. And then under Organize, it says Add Shortcut. And then I can choose to put it in my drive. And once I've got it in my drive, that shortcut in there, then you should be able to get to it from Google Colab. So let me run through that again. I've got a folder that I want to put and get access to for Colab. Select it and then go to the triple dots on the far right end. Go to Organize, then Add Shortcut. And then I tell it I want to put it in my drive. And then I should be able to get to it from Google Colab. All right, so hopefully that helps.
All right. So what I'm going to do is I'm just going to click on this notebook. This is on my GitHub repository. When you click on the link, you should see something like this. Before you open this notebook, you might want to go back and close the other Colab notebook. Because what happens is if you have too many notebooks open, then you might get a session timeout. So please go back and close notebook 1. You can save it first and then close it. And then once you've done that, click on the Open in Colab button. And here is our second notebook on basics of Python syntax. And these notebooks, you have access to these. So go in there, save a copy to Drive. When you do that, it opens up a new version. It's called Copy Of. I'm going to call it Crash Course April 30th. Okay. And now it will be in my drive with that name. And so what we're going to do in this notebook is we're just going to use some standard Python materials or Python commands to do a variety of things and to just get familiar with how Python works. In later notebooks, we'll be looking at libraries, which are collections of functions that you can use. And so there's no we're not going to add any libraries this time. And so the rules about running in Colab are the same as they were before. So I don't think I'm going to review this, just the idea that this is the run button. We don't need to set up an environment or libraries for this notebook. We might need to do that. And for this notebook, I'm going to import my Google Drive just so I have it. So I'm going to open the folder window here so you can see what's going on there. And when I run this cell, notice there's a dotted line around the run button. That dotted line tells us it's basically getting connected. And once it's connected, and then it shows a solid line circulating. And then I go through and I give permissions. And now it's connected to my Google Drive, and that should show up over here fairly shortly. Notice there's a command here, drive, flush, and unmount. So what that does is that clears any drive that I've got loaded. And now it says mounted. The drive is now mounted at content drive. So it's going to show up in this window soon. It's not there yet. Oh, there it is, drive. Okay, so it's there. Now, I'm not necessarily going to do this. What we're going to do in this section then is we're going to navigate and execute code in Google Colab. And we're going to look at basics of Python syntax, variables, data types, and some operations. And one of the questions you might have is, what is Python and why is it useful for biochemistry and molecular biology? We'll see kind of a hint of that in this notebook. We'll see more clear demonstrations in notebooks 3 and 4. And we've already gone through what Colab is and how it can be used to run Python code. So the learning objectives here are just to learn the basics of Python and Colab, to work with variables and data types, to use for loops, and to use logic statements. So if we go to section 3, we've got a cell here. Now I've got my line numbers turned on. So line 1 says 3 plus 9. Now this is a coding cell. I can tell that because it has the run button on it. Now I can run this cell by clicking on the run button, or I could hit Shift Control Enter and it would run. Okay, so if I run that, you'll notice when you run it, you'll notice that it gives you the value 12. So you can use the run cells as simple calculators, but for coding purposes, you want to assign values to variables that you can use. And so the way we do this is we have a variable name. We say variable name equals, and then we give some value that we assign to that variable. That value could be a wide variety of things. We'll be looking at integers, which are whole numbers, at decimal numbers, we call them floating point numbers here, and also strings, which is just simple text, all right? And so what we're gonna do is in the next cell, we're gonna declare some variables and parameters and do a simple Michaelis-Menten calculation to determine enzyme velocity. So here's our problem. What is the reaction velocity if the Michaelis constant for this enzyme is 15.0 micromolar? The maximum velocity is 100.0 nanomoles per second. The substrate concentration is 8.0 micromolar. Now, I've got a comment here, calculate using the Michaelis-Menten equation. So if I go to this next line, watch what happens, and you watch what happens on your computer. You type in Km and see what comes up afterwards. All the information you had in the cell before shows up. And if I go here and I hit the tab key, then it becomes solid text, and I can run this cell and get a result. Go ahead and do that. Now, what I want you to do is hover over the bottom of this cell. There's a option for code. So we're gonna open a code cell again. So here's another code cell. Now this time, I'm gonna change it a little bit. I'm gonna say Km equals 15.0, and then I'm gonna put in pound micromolar, and then I'm gonna see what it does with that. Notice it puts in the remaining information, but now it's added comments on the lines that have variables. And so this is part of the way that Gemini is participating in this process. Okay, so Gemini will help you with your computing. And it's always important when using Gemini. I really like what Wally said before about having students write it out by hand, what they're going to do, and then do it with, and then have Gemini help them do it. And if I run this, I get the same answer. All right. So notice that when we put a pound sign there, the information after the pound sign, it's useful to us as the coders, but it's not part of the calculation. All right. So that's important. Also, when we choose variables, it's useful to choose names that mean something. I could have just put in S or capital S for substrate concentration. Maybe that would have been okay. But it's better to use variables that are meaningful, but not too long. I try to make them 8 characters. This one's 12 characters. All right. Now, we also notice we've got this print statement here where we say print velocity. So with this print statement, whatever I put inside here, it's going to print. Now, if I have a variable with some value assigned to it, it's going to print it. And so it just prints the number again. Now, what I want you to notice in the next cell is I'm going to tell the cell, the code says print velocity. Then it says velocity equals velocity times 60, and then print velocity again. So what we see when we do this, when we hit the run cell, what we see there is that the value for velocity has changed. We've overwritten a variable. Now, there may be times when you want to do that, but it's usually better not to do that. It's usually better to write a new variable to meet the thing you're trying to do. And so what I'm doing here in the next cell is, first of all, I'm going to change the velocity back to where it was. So I take this velocity where I multiplied by 60, I'm dividing by 60. So I end up with a value that actually is going to match this original value. And I'm going to create a new variable. I'm going to call it V nanomoles per minute. And then multiply velocity by 60. And I print velocity. And then I print the V nanomoles per minute. And so once again, I got those two values. but now these two values are assigned to two different variables. All right? So what I want you to do, very simple exercise, I'll give you just a minute to do it. In the code cell below, create a variable called km2, set its value equal to 30, and then print that value.
Hi, Paul, while they're doing that, there's a couple questions. Can you go back up to the where you were entering like the KM? So some are not getting an autofill for like the KM based on that.
Okay, I'm not sure why that is. Maybe it's the fact that I've run this notebook a bunch of times, and Gemini knows what I'm up to. Now, when I did the second cell, I filled in the first line, typed in micromolar, and hit return, and then it filled in the rest of the information. So once again, as I said, I've done this many times, and so maybe it knows that I'm doing this, and it reads it, and it knows what's coming. Do you have a better answer than that, Wally?
No, I think that might be it. And so really what they would need to do is just kind of is type some of those things out. Is that correct? Like the KM equals and then just hit return and Vmax equals, which is just
Right,
the information is up in the statement above it, the text box.
right. So you may have to manually type it in, and I think there are actually settings that you can go in and change so that it does interact more with you in that term. And I'm not sure what the defaults are. All right, so just to demonstrate how this works, we're just going to say km2 equals 30. So there's our second km value. We're just going to do print km2, and it prints out the value. Okay, so next we're going to look at what kind of data types we have. And the most common types we use are strings, which is represented by the letters str, integers represented by int, and floating point numbers represented by float, okay? And the way you find out what kind of variable or what kind of data is in your variable is you use the type function. So you type, and then in parentheses, you put the variable. So if I type and I look at velocity, it tells me it's a float, all right? Now, if I come down here, I can actually take that number and assign it to a string. So I create a new variable called velocity underscore string. And I'm going to say that's going to be equal to the string version of the number that's contained by the current, by velocity. And then I'm going to do a type of velocity string. And what we're going to see is that it's a string. All right. So what I want you to do in the next cell as an exercise, give you a minute to do this one. I want to convert velocity to an integer and then print the result. And like I said, I'll give you a minute again for this one. And the question about autofilling, if we go to Tools, Settings, AI Assistance, you can check these boxes for Show AI-powered inline completions, consent to use generative AI features. So those are a couple options you can select, and I've selected those. So maybe that's why they weren't filling in for some folks. All right. So what I'm going to do is I'm going to say velocity int equals int velocity. And you can see that Gemini did that for me. And we're going to print, well, we could print that, but what I really want to know is the type of velocity int. So if I go through and do that, it tells me the number is, notice it truncated the number to make it an integer. And I'm trying to see if it rounded the number. The value before was 34.78. It did not round. It just took the integer portion. And now the type of that velocity int is an integer. I'm glad to see people putting questions in the Q&A box, and I know that Wally and Chris are following that. Now, another common data structure we use in Python is the list, okay? And lists are in square brackets. Python is very particular about brackets. So square brackets contain lists. Parentheses are shown here. Those are often used if you want to assign a variable to have a function act on that variable. So we saw that, for example, when we say print velocity int here, we've got within the parentheses, we've got velocity int. So that's typical. And then curly brackets are used for a number of things, particularly dictionaries. You need to be very careful. These are not interchangeable, all right? So what we're going to do here is, first of all, create a list. So I've got a list, I'm calling it, you can see it's substrate concentrations. I didn't write out concentrations fully, but with the S there. And you can see that I've got an open bracket here and a series of numbers. And then I've got a comment after that saying micromolar. Now, it's possible when you have a list that you can find the length of that list. So if I want to find the length of that list, I use a function called len, L-E-N, and then I just put the variable, which is a list, in the parentheses there. And so I'm going to say S length equals the len of substrate concentrations. And then I'm going to print the list, but I'm going to get a little fancier on the print. I actually want the print statement to provide me with context. So I'm going to say print, and then in quotes, this list contains, comma, S length, which is the variable, doesn't have to be in quotes, and then in quotes again, substrate concentrations. Now, notice that I've got the text that I want to print in single quotes. As far as I understand, I can use single quotes or double quotes interchangeably here, but they just have to match. So if I run this cell, you can see this list contains 13 substrate concentrations, all right? So we can use the print statement this way, just to say print, and then we can put text in quotes, variables just as the variable itself, and then text in quotes after that. There's also something called an F print statement, which is very nice, and I like them a lot. So I can write print, and then inside the parentheses, I put an F, then I put a quote, and I enter my text, and then any variable I want to use, I enter in curly brackets. Then I enter more text, and then I enter another variable in curly brackets, and I can do that as many times as I want in an F print statement, and then I close with a quote mark. Now, the nice thing I like about this is that I know exactly the spacing I'm going to get every time, all right? So if we try this print statement, this list contains S length substrate concentrations. That's actually in the next cell. If we run that, you can see that it's exactly the same print statement we saw up here in terms of the output. Well, I guess this one doesn't have a period after it, but it's the same print statement. But I really like the F print because it helps with formatting. All right, so let's go back to substrate concentrations. So now it's possible to convert the last substrate to address one of the elements in a list. We're going to do that here. We'll get a little bit more to lists later. But the 12th element in that list, we can actually change the concentration of that from micromolar to nanomolar. So we're just going to do that on one of the elements in that list. And we multiply it by 1,000, and we will see the nanomolar concentration of that one becomes 10,000 or 100,000 instead of 100. All right. And if we look at the list, which is up here, we can see that there are 13 items. So the first item in Python, you count starting with a 0. Okay, so the 0th item is 1. Then item 1 is 2, 3. I'm sorry. This 2 is item 1, item 2, item 3, item 4, item 5, item 6, item 7, item 8, item 9, item 10, item 11, item 12 in the list. All right. So if I go in here, it's acting on the last item on that list. Okay. Now it's also possible to add something to a list. So our current list has a highest value of 100 micromolar. Well, I can add a value at the end using the append function. So I take my list name, which is substrate concs dot append, and then in parentheses after that, I put a value that I want to add. When I do that, then you can see that my list is longer and it has an additional element on it, 100 or 1000. So what I want you to do now is I want you to print the last value in that list. You can print that, and we'll look below. We will look at little word lists at how this works. We can use this statement: print substrate CONCS, and then in the bracket here we say we're going to take the length of that list. We're going to look at the last element in that list. Okay, and we subtract 1 because remember the first element in the list is element 0. And then I want you to add the value 200 to the end of the list with the append function. So I'll give you a couple minutes to do that. Okay, so I realize now that all you really had to do was copy and paste the materials into the line. And so I just did that. And so I found the value for the last element in that list. And then printing out the entire list, it's shown here. Okay, and you might look at this and think, well, that was pretty simple, Paul. Well, it was pretty simple, but we want to show you simple things to get you more comfortable with the idea that coding can be done. All right. We're going to look at slices for just a minute. So a slice is a part of a list. And this is a tool you might want to use in a variety of settings. We'll actually take a look at using this later. But we can create a new list from an earlier list by telling it what part of that list we want. So for example, if I say short list equals our earlier list, but only elements 0 through 3, or actually it's 0:3. So when I run this, I've created that short list. So that new short list contains element 0, element 1, and element 2 from the original list, but it does not contain element 3. Okay, so it goes 0, 1, 2, but it doesn't do 3. All right, and we'll see that if you hit the print short list button, then you see we've got the 0th element, the first element, and the second element. And I'm actually going to skip over this next part because I see that my time is running a little short. So we're going to move on to for loops. So a for loop is something you can use to repeat an operation many times. The syntax for a for loop is you type in for some variable in some list, follow that by a colon. When you hit return, Python automatically indents the next line by 4 spaces, and then it does things to whatever variable you put on that list. All right, so here I've got this list, substrate concentration. So I'm back, I've redefined my list as the original 13 elements, okay? And what it's going to do is for each number in this list, substrate concentrations, it's going to calculate a velocity based on the Michaelis-Menten equation and print that velocity. And so when you hit run, that's what you're going to see. And each time it goes through, it prints the value. And so we get 13 values for velocity as a function of substrate concentration. And Now you might think, well, I'd really like to have those values in a list. I'd like the velocities in a list as well. So what I'm going to do now is I'm going to have basically have a list called velocities and add the values of the velocity that we calculate to that list using the append function that we saw before. So I hit the run button here. When I do that, I get an error. Okay, what's the error tell me? Well, as I understand Python, the best place to start looking when you get an error message is at the end. Go to the end, and at the end it tells me velocities is not defined. Okay, now I'm here to explain this to you, but if I wasn't here, you could click on this button and would provide you with some more detail. Gemini will help with that. Okay, so what we need to do is, first of all, we need to create an empty list called velocities. And there's nothing in it. And so what we're telling this for loop is going to do is it's actually going to end, put the values for the velocities. It's going to add each one as it's generated. And then we come out of the loop. So we're back at the left-hand margin, I guess I would say here for line 7, come out of the loop, and it's going to print that new list. And so here's our new list with with the 13 values that were calculated. So that's a separate list. And I don't know if you noticed before, but there's a function called zip that you can use to pair two lists with each other. We're not going to do that here, but I think you're going to see that later in one of the other notebooks. All right. So once again, looking at the time, I'm not going to have you do this exercise to print velocity on its own line in light of the time. Okay. We are going to take a look at logic statements. Okay. So we use if statements to do logic, to work with logic, and this is how we make decisions in a process. Okay, so now as we're doing these calculations with velocities and substrate concentrations, you're a biochemist or molecular biologist. You want to plot that stuff, right? We'll get to plotting later in the workshop. Okay, so here I've got a statement or a series. We've got a variable temp. We have a value of 35. Okay, so that's the number we want to check. And then we say if temp is greater than 30, then it's going to print hot. And so if we run this cell, we see that it does print hot. Now, if I change the value now to 30 instead of, if I change it to, let's make it 25. If I change it to 25, 26, and I say, and I run it, doesn't print anything. There's no output. So we're actually going to use a for loop to print the results of some calculations by, we're going to actually use logic within a for loop to separate a part out. So let's say we want to look at the linear part of a Michaelis-Menten curve. So that's the point where the substrate concentrations are less than 1/4 of the Km value. And I'm not sure if I think that's a linear range. Anyway, a couple things to notice. The first statement here, Km comma Vmax, I'm assigning two variables in a single statement. You can do this. So with this statement, the value of 15.0 is assigned to Km. The value of 100.0 is assigned to Vmax. I create a new list, it's empty, called linear MM for Michaelis-Menten. And I've got my same substrate concentrations. So I have it go through this list, the values in the list substrate concentrations. And if number is less than 1/4 of Km, then it's going to calculate the velocity, and we're gonna add those values to our list, linear MM. And if we do that, you can see we only get 3 values here. Well, if we look at the Km, the value is 15. So any number less than 3.75, the calculation is going to happen. But if it's more than 3.75, the calculation wasn't done. Now, it's possible to use other operators as well for this. Equals, if you want to see if two numbers are equal to each other, we actually have equals equals. So we're actually comparing them with each other, okay? The exclamation point equal means not equal. Greater than and less than are obvious. Greater than or equal to, you put the greater than first and then equals, less than and then equals. You can also use and, or, and not to do Boolean type of logic for this. And once again, I'm almost out of time. So I'm going to skip over this, but just to mention that we've got a Boolean here where we're comparing we want a number that's either less than the km or equal to the km, and doing a calculation there. And I've got some additional things to do, just to writing if statements. Actually, let's go ahead and do this one. So go ahead and write an if statement that will print this out. And I want you to notice when you put the colon after the if statement, how the indentation happens. Okay, and this is one way you could code it. And if I write that, then it works. And if I change this to 35, then it doesn't print. Great, that makes sense. One thing I want to point out as I'm wrapping up is that you're going to notice that Chris and Wally and I do things a little bit differently from each other. There are some practices that are very good to follow. We do our best to follow those, but we have our own preferences about other things. And so just as in the same way that you probably teach, if you teach Michaelis Menten kinetics, you might teach it a little bit differently than I would teach it. You still have the same basic things you want to get across, but there's style, your personal preferences. We have an exercise here. You can work on that during the break. I'll come back at 3:25 and actually take a look at the solution to this exercise. So for this notebook, we've looked at calculations with variables, different data types in Python. We created some lists and sliced them. We ran a for loop for repeated calculations, and we used some logic statements. I've got some links in this notebook for a number of resources I find very helpful. The Home of Python is just the place where you can download Python, and there's a lot of resources there. Problem solving with Python is actually a textbook. You can get a paper copy of that from Amazon, but the entire book, including solutions, is online. And this is where I learned a lot of my Python coding. Corey Schaefer has created some marvelous Python tutorials. This is a link to his YouTube channel. And there's an exercise here you can do. There's an optional challenge on the bottom that you can do. So you're welcome to do those things. All right, so the problem once again is we get a list here. And notice this list contains strings. So 1.0 here, it's in quotes, so it's a string, it's not a float. Okay, so 1.0 and 14.0 are strings, not numbers. Okay, so we're going to write a for loop. And if an element is a string, we're going to recast it as a float. Okay, so we have our list here. We create a new list we call number list. It's an empty list. And then we're going to go through that list. So for each item in the list, we're going to ask if the concentration of type of an item is str, is string, colon, don't forget an indent here, item equals float item. So it's recasting it as a float. And then it's going to add that number now, that item, which is a number, to the number list, to append it, okay? Then, now a couple things, and then it prints out the number list and we get the answer we want. A couple things to note here is, first of all, words like in, you notice they're a different color, is, different color. Those are reserved words. You can't have a variable called in or a variable called is, okay? Another thing to notice is they used is here as a comparison term. You can also use equals here, and it does the same thing. It serves the same function, all right? So those are just a couple points to make there. And I'm going to stop sharing And at this point, I'm going to turn things over to Chris.
Getting my screen shared up here. And I do want to say that I appreciate all of you hanging in here and working through this. I know you've probably gotten a lot thrown at you today, and if you haven't done a lot of coding, they can seem overwhelming. And certainly when I started really getting into coding about 10 years ago, maybe a little bit before that, this would have hurt me as well. So please do hang in there and let us know how we can help you. And we will try to do our best to explain things as best we can and keep you on track. Um, we will not judge your code. We will only try to help you make it work. So in this third collab, we'll be working with something called Pandas. And so Paul has had you been working with lists and strings and numbers that you've typed directly into the code block. Pandas allows you to start working with numbers and data more like it's in a spreadsheet, which is traditionally how I would think about working with data, or when I'm getting data off of an instrument, it would come down as a series of columns and rows, and it might be large, it might be small, but I'd have data in a two-column format. And so Pandas and NumPy are really good at working with those type of data and organizing data that way as well. So we're going to start the collab like we did before, but I'm going to ask you to do one thing. So I'm going to go over here to the top right, and it's going to say Manage Sessions. It's in the third section. It's the second option there. If you click on Manage Sessions, it's going to show you all the different collabs you have running. You may want to X out of the ones that are not the current session, just because you can sometimes hit a timeout error, or sometimes collab gets really angsty if you have multiple sessions running. So I'm going to close the session that I was running earlier and get that shut down and only work with the current one here in Pandas and NumPy. Now, I've already connected to this because I was making sure that something worked and trying to help someone answer a question earlier. So I've already run this first block, but I'll do it again. And it's going to import NumPy, Pandas, and then two plotting programs, Matplotlib and Seaborn. We won't work with these a whole lot. We'll do a little bit with it, but we will. They will be in the challenge section at the end of this collab if you want to play with them a little bit more. The other thing to note is that when we refer to using Pandas, we're going to call it PD. It's a shortened way when we're typing code. And when we refer to Matplotlib, we're going to call it PLT, or Seaborn, we'll call it SNS. And I'll show you examples of where that's going to come up for us. But we're going to use shortened versions of these packages so we don't have to type out everything all the time. Now, in Paul's collab, he was writing out numbers like this, and he gave you key values to assign variables. And when you run that code block, it doesn't produce anything, but it stores those values so that you can use them later. And then this is one of the for loops that Paul had you make, where it goes through and calculates out calculates out Vmax and Km, fits the data, and gives you a set of values here. So we've got this long string of values. And if we look at what type of data we're dealing with in both velocities and substrate concentrations, these are lists. And lists are great in that you know the data, you have it in a specific order, you can see it all in one place. But again, it's not often how we get data or how we collect data. Think about if you're doing a spectrum of a small molecule, or if you're collecting CD data or something else. You might have absorbance in one column of the output from the instrument along with a particular value or a particular characteristic in another column. And so that's where having lists is limiting, and that you aren't connecting the data together. Velocities and substrate concentration have no relationship. They are just lists that we use together, but they are not something that you have a connection like we would in a spreadsheet. One way you can do this is what's called a tuple, and we won't go into this too much, but a tuple is a way to store data and keep pairs together. But you can't do things like sorting and grouping and some of the analysis that we might want to do. So if you have values that you want to keep together and do not want to change, a tuple is the way to go. But we're going to work with what's called a data frame, where we're going to put data together and have them in a 2-column format in this first example, and then we'll work with several columns later on, just to organize it in a way that I feel I'm certainly more comfortable working with. So to do this, I'm going to give it a name. I'm going to call my data frame DF, and then I'm going to call pandas. Remember, I said I was going to shorten pandas to PD, and then tell pandas what I want to do. I want to make a data frame, and then I've got my function where I've got my parentheses, and within those parentheses, I'm going to put the curly braces with substrate concentration, which we made earlier, and then give it a name, give it a column a name, and then give it the name of the list where that is, and then give my second column velocity and tell it where to find velocities in the velocities list. Once I assemble that, I'm then going to print that data frame out in the window. And so that took less than a second, but I have my standard 2-column format that I'm way more comfortable working with as a biochemist, in that I know how the data relate to each other. And then if I wanted to add other columns or do sorting or fitting or put this into a plotting program, I could do that more comfortably from my perspective. So let's work with a different type of data frame that's going to have some diverse characteristics and try to explore data frames with pandas in a little bit more detail. So we're going to make a data set that's going to be a plant growth experiment. I work in plant systems, and so I think about species and watering and fertilizers and temperatures and things like that frequently when I'm doing work with the Arabidopsis in our lab. And so we're going to make a synthetic data set that is going to have these different columns, and then I've got a descriptor here to tell you what is in there. So this is like a README for my data. And for those of you who might be sharing notebooks with students or might be sharing data with students within a collab, having a table like this at the top is really helpful for students. They really appreciate it if you give them a guide to the data and just give them a hint about what they might be seeing. I'm going to create the data, but I'm going to hide the code from you all. So if you click play, it's going to run and create a 200-row data set with 9 columns. If you want to see how the code works to make the data set, you can click show code, but it's not relevant for what we're going to be doing. So I'm just going to operate as if the code is hidden, and we've got our data set. Now, if I had a new data set and I wanted to understand a little bit more about what I was dealing with, maybe I want to look at how the data are structured or know the column names of the data types in each column or just general statistics. You can do that with pandas. And the way that we do this is we tell pandas what data to work on, in this case, DF, which is our data frame. And then we put a period and then tell it what we wanted to analyze. So I've got 6 common functions here. One that looks at the first few rows, the one that looks at the last few rows, one that will tell you how many rows and columns you have, one that will list the column names, one that will tell you the data type, and then one called info, which is a compact summary of all of your data. And so in these next couple of blocks, let's explore the data together. So I've got my comments here telling you what to do. And for these types of exercises, I usually put in some sort of space or a gap to tell you where to write the code. This is especially early on useful for students and new learners just to know where they need to put things. So in this first block, I'm going to type df dot, and then I'm going to type, because I want to know the very top part of the data frame called the head, I'm going to tell it to look at the head. So I'll type head here. And I only want to look at the first 5 rows. And so there's a 5 in the parentheses. And I'll run this, and it prints out the first 5 rows of my data frame. And if we had a few more columns, we would need to slide. If I wanted to look at the first 7 rows, I could change the number to 7, and it will print out more of the rows. So this is one way to start looking at the data. Why don't you try to go through and take these commands from the table above here and put them in where I've got the spaces and try to do these next steps. So I'll give you all a minute or two to start filling in some of these, and then we'll go through them together real briefly and go from there. right. Now, when we're doing these different commands, and I know you may not be fully done, but I wanted to say that some of these commands are a little tricky because some of them include parentheses, like head, tail, and info, and some of them do not. And so this is where students get tripped up, is remembering which ones to include parentheses. And as an aside, I often forget which ones need parentheses. And so it's common that I need to go look it up and make sure I know which ones need that particular set of information. So if I want to look at the column names, I'm going to type df.column. And it then says, oh, it's actually not column, it's columns. So I'll click on that and it will give me the columns that are in this particular sheet. But when I get the data types, I can go in there and type D types. And you'll notice I'm starting to see an autocomplete function. I'll show this again just for those who may not have seen it. So I'm starting to type command. It gave me the function that it thought I wanted to do. And I was able to select this with my mouse, or I can hit return and it'll pop up. And now I can get a list of the data types. And then the one that is often very useful is the info command. So I'll click on info and it goes through and tells me that for some of these columns, some of the data, I have 200 bits of information, whereas for some of them, I've got a few less. And then it also tells me the data type over here as well. So when you have a new data set, it's often good to look at how it's structured using head or tail. And then I like to go in and look at the info just to see how complete my data sets are and the data types and to know the columns overall. And sometimes I'll even take a screenshot of this and have it sitting in the corner while I'm going through my data just to keep track of things. All right. Well, we know what our data look like. Now let's think about doing some statistics on this. Just like in Excel, where we can go and highlight some data and say average or standard deviation, we can do similar types of things in Pandas and then also in NumPy. We're going to focus on the Pandas functions right now, but I give you the correlated or the analogous one for NumPy in the right column of this table. And you can go in there and say in the data frame, I want you to calculate the mean values or the medians or the standard deviations. And so let's try that in this first setup. I've already gotten written out. It's data frame or df.mean, and then I've got the parentheses. I click on it and it fails. And I could explain the error using Gemini, but down here at the bottom, it also tells me that it could not convert a bunch of letters into a number. Well, that makes sense. We can't take a mean or an average of ABC. And so this will not work on it alone. So there's a way around it initially that we can do if we want to look at the entire data frame, and that's to use describe. So describe is shown here, and it gives the basic statistics in one command. And so we click on this, and it ignores the column that had the ABC in it, but all the other numeric columns, it tells me how many I have in how many rows that I have that are not null, the mean, the standard deviation, the min, the 25, 50, and 75th of quartiles, and then the max value that's in there. So this is useful for just looking at a data set and trying to understand what is going on in there. Now, maybe I don't want to look at the entire data set. Maybe I've got hundreds of columns and thousands of rows, and it just would be overwhelming to do. Maybe I just want to look at a single column. Well, we can modify our commands to not do it on every single column in the data frame, but to do in just one. And the way we do this is by saying which column we want in brackets and then having the column name within the quotes. So what I'm going to do is I want you to calculate the mean value for the species in the column following this example. I'm going to tell you that this first one is going to fail, but I want to see how it fails. And then the second and third code blocks, use the describe function or then define the column by filling in the blanks to or these statistics a little bit. So I'll give you all a moment to try these out. Remember, the first one will absolutely fail on you, but it's worth looking at it. And then the second and third ones will work. They will give you values. You just need to fill in the underscores, the blanks there to try to get the right values in there. So I'll give everyone a moment to try that, and then we'll work together to make sure we've all got it clear. All right, so remember I told you that first block was going to fail. It's going to fail for the same reason that we saw earlier. We can't take letters and convert them to numbers. But it's worth seeing that error, and the errors will always tell you what the problem is at the very bottom there. These can be a little bit dense, especially if a really complex function, but for something like this, it tells you pretty clearly what is going on. You can then copy this into Google if you want to search. If you don't want to use Gemini, I frequently just copy these and put them straight into a Google string, and it'll give me some clarity as to what's going on. Certainly, you can also click explain error, and it will not only tell you what the error is, but also tell you how to fix the problem. All right. So the second one here, I wanted to use the describe function, but only on leaf count. So I'm going to replace this blank with describe and then the parentheses. I'm going to run it. And rather than the long or the really wide data set we've got here, I've really just shrunk it down to the single column that I was interested in. And it gives me some nice statistics here. So now I'll do the mean value here. Now I've got the function and I know what data frame I'm doing. Now I want to add in the brackets, and then in quotes, I will type in water mill, and then it gives me the value, and it puts it within this NP float 64. So it tells me that it's a number with a lot of decimal places on it. And we can get rid of that by, rather than just saying the value itself, we put that inside of a print statement. And so Paul showed you some of these earlier, but you put print and then you put what you want Colab to print, in this case, the mean value for this particular column in the data frame, and you will get the number. Now, there was a question in the Q&A earlier about integer and then it not rounding right. Well, one way you can round things is you can use the round function. So I'm going to go back and add on .round and then parentheses. And maybe I want to round this just to one decimal place. So I put 1 within the parentheses. So I've got .mean with parentheses with nothing in it. And then I do .round with a 1 in that parentheses. And I run that. And now I've got a number that has been rounded to 1 decimal place. You can certainly change this number and it will alter it and round it appropriately based on the number that you give it. I use this and really I tell students that they need to be careful with their significant figures. Being a chemist, we've got to have the right number of decimal places or values in here. So round is useful for getting those values right and not having that string of numbers that you often see in Excel. And Paul showed you how to make F statements. So maybe you want to make this notebook something that speaks in more human text, or you want to include the units or something like that. So let's go in here. We can also incorporate these values into print statements. And the AI is telling me what I need to be including in this case. Now, one thing that is missing from this is units. So I'm going to go in here and edit this and say milliliters, just to add that in there. And when I run it, the mean value for watermills is this, and it gives the units in there. You can adjust the number of decimals in this particular F statement by that dot to F. If you change that number to something else, it will change the decimal places that appear in that statement. So it was 2 in the default for me, but you can go in there and change this. And another question that I got earlier was, will these notebooks, is there an answer key? I know for mine, I'll put some answer keys, or I'll put the filled out ones into GitHub for you all to look at later to see if you can recreate the code if you have any questions about it. But I don't know if Paul or Wally are going to do that. But for mine, I'll try to put them back in the GitHub for you. All right. So we've gone through and we've started being able to calculate values and pull together values from statistics, from single columns and from areas of our data frame to analyze it. Maybe we just want to look at a few columns together, not one at a time, not the whole spreadsheet, but maybe we want to look at a couple of columns. We can do this by creating a list of our column names, and we create the list within brackets, and then we have quotes, and we separate the column names by commas that exist outside the quotes. So in this case, I have light hours, comma, watermill, comma, fertilizer G, comma, and leaf count. And then I have the brackets on either side. And then I'm going to create a data frame from this. So a whole new data frame that I'm going to call stats. So I have stats equals pandas. I remember I abbreviated that to PD dot data frame. And then I have my open parenthesis, open curly brace. I put in the names of the columns in quotes, and then colon, and then what I want that value to be, just like we've been doing previously. But this time I'm not going to put specific column names. I'm just going to put my list name. And then I close that up, and then I'm going to print it out down below. So take a look at this code, make sure it makes sense to you, and then run it. And you should get something that looks very similar to this. We have lots and lots of decimal places, so maybe I want to change that. So I'm going to go back in here, put round in there, and maybe I'm going to round it to 1 in this case, and I can do that. So now I've taken the stats data frame and rounded everything that I got out of it to 1 decimal place all at once, and it makes something that's a little easier to read and not overwhelming with the number of numbers in it. All right, let's take a moment. If you need to catch up or take a breath or go back and review something to make sure you understood it, take a moment, and then we'll move on to grouping in just a moment. So I'll take a second here to slow down and stop and let everyone review as needed, and we'll go from there. All right. In this particular data frame, we have our numerical data, but then we also have data on 3 different species, A, B, and C in this case. Well, maybe we want to compare the species to each other. And so we would want to maybe look at the mean, median, standard deviation, and the number of values for each of these species. We can do that using something called group by. And what that does is you define 1 or 2 columns, and sometimes even more if you really want to get complex, but you can create subgroups within your data and then start pulling out individual numbers for those groups. And I find this is one of the most useful functions for Python, and I also do this with R a lot, in that I'm able to put a massive amount of data in, maybe for different enzymes or different species or different days or different students, and then pull their data out as a subcategory, but compare it. And I don't have to go through and sort an Excel spreadsheet and then do all the individual calculations. I can do it in one long line of code. I've got that code broken down here on in the text box, and then I've got the code written out here on line 3 of the code block. It's fairly long, but it has some clear things that it's doing. So I'm going to say, tell my data frame that I want to group by, and I want to group by species. Then I indicate within that species group what columns I'm interested in. So I have two sets of brackets here. I've got my column names in quotes separated by a comma, and then I'm going to aggregate them and calculate the mean, median, standard deviation, and count. And in the end, I want to round it to 3 decimal places. So in one line of code, I'm going to go through 200 rows of data, create 3 groups, species A, B, and C. I'm going to look at the height and the leaf count and compare the statistics on those 2 columns rounded to 3 decimal places. I'm going to run that, and I get this nice looking data frame that has the mean, median, standard deviation, and count for height in centimeters, and the mean, median, standard deviation, and count for leaf count, and the rows are my species. And so when those of you who are thinking about big data sets, or if you want to compare multiple data sets together, this is where Pandas really is powerful, is that you can take these massive data sets and in seconds create these wonderful comparisons that are clear. And effectively, this is a separate data frame entirely. And so you haven't manipulated the original data. You've got a new data frame, which is a summary data frame of your data. We can also do some really simple plotting. We're not going to go into a lot of the details. The challenge questions do a little bit. We're going to keep them simple here. If I want to do a histogram, I say df.hist, and that'll create my histogram. And then I type plt for plot and then show. So I tell it what I want it to make, and then I tell it I want to make the plots and show them here in the frame. So I click on that, and it generates a histogram for all of the numeric columns in my data frame. If I just want to do a single column, say we want to do height, I can go and put brackets in like I've done before, type height cm like I've done before as well, and rerun this, and it creates a single histogram for me. So we can also do multiple histograms. Let's see, was it leaf count, I think is the other one. Oh, it's not going to let me do it that way. Oh, I bet you I remember why. See, sometimes we make mistakes. We get adventurous and think we want to do it, and then it's not going to work this way. I'll figure out how to do it and put an example more for you. We'll skip over this and go into the categorical variable section. All right. So categorical variables are ones like species. We don't have a number. We can't do means and medians, but maybe we want to know how many that we have. We can use something called a value count, which will go in and count the number that fall into A, B, and C in this case. And then we have our parentheses, and then we're going to print it out. So I run it, and I get my species counts, and it tells me that I've got 82 plants in category in species A, 69 in B, and 49 in C. And so again, it's a way to go in and look at specific columns and categories. Now, very early on, when we looked at the data set, I did a first look up here at the top. We said that we had some columns that were missing some data. So our water in milliliters and our health score, they didn't have 200 observations in each category. They had somewhat less than that. So we can go and remove those data, or we can go and identify those data using some of the code below and understand which categories are missing the values if we haven't done this already, but also to calculate how much of our data is missing. And I've got some example data written here. If you run these data, it will say that in health score and in water mills, as we knew, we're missing some observations, and that represents around 5% of the total data in there. And you could make a decision about whether you wanted to include those data or remove them. And if you want to remove them, you can drop them using the drop in a function here. All right. I know we're coming up right against the time, but I do want to show you how to make new columns, because this is, again, where Pandas and these types of programs are useful. In Excel, sometimes we do mathematics, where we would take substrate concentration and velocity and calculate AV max or a KM or something like that. Or maybe we want to multiply columns or create a ratio. You can do that using much of the nomenclature we already know, where we define our data frame and say the column, and then do our mathematical function, whether it's multiply, divide, addition, subtraction, logarithmic functions, whatever. And you create a new column simply by saying data frame and then defining the new column name. So that's what I've done here in line 2. And then if I wanted to create a list of columns that I wanted to print and see what this looked like, I could say, well, I want to see height in centimeters, leaf count, and height per leaf. And then if I run this, I get my data frame that shows me not only the 2 original columns, which are height in centimeters and leaf count, but also the new column, which is the height per leaf, which is the ratio of those 2 put together. Again, you can do a lot of cool math with this, and I've got some exercises here down below for you all to try later on. The last section is about importing data frames and how to do that. We've done that a little bit, and I've got an example here using a PD for Pandas and then read CSV, and then I've got the GitHub link directly pasted in here to create the data frame, and then it prints out. So you can try that. The last 2 sections are, I've got how you can do these types of statistics in NumPy. It's a little bit different than Pandas, but it still is, it still will give you the similar answers and give you the statistics. And then I've got some challenge questions down here for you, where you're going to be working with some pKa data from a molecular dynamics simulation and working through that and trying to do some of the things up here above. Again, I've got an answer key for this for those who want to see it, and I'll put that up on the GitHub for those who are interested. But I hope that by going through this, you have a sense of how Pandas can be useful for you, especially with big data sets, and be able to process data quickly. And we will then, I believe, are taking a break after this. So I'm going to stop sharing, and Paul and Wally can keep me on track if I've fallen off. So.
Hopefully everyone can see the screen. Again, just if you can close out everything else. I'm just sitting here now on the CodeBMB site, and I'm going to click on Notebook 4, Linear Regression. And I'll click Open in Colab. And then what I'm going to do is do File, save a copy in Drive. And that's going to pop open a nice copy that I will just play with. And if you follow those instructions, you can do whatever you want in here and save it. It'll just save right to your Google Drive then. So in this notebook, we're going to just run through some linear regression, right? It's a really common thing to do in biochemistry. It's going to draw some of those skills from the previous notebooks. I actually want to do one thing before I reminded myself. I'm going to just turn off my AI assistant, because that way, if it doesn't pop up for you, no worries. You can leave it on. But this is an overview. We're going to load some experimental data, which Chris showed. We'll calculate means and standard deviations across replicate measurements, build some scatter plots with error bars, and perform a linear regression. And we'll look at how to look at some of the documentation as well. And then there are some challenge things that people can tackle at the end. So we do have some input data. So this is just a CSV file. For those of you who are not familiar, a CSV is just comma separated variables. And if you're in Excel and just on a single sheet, you can always save your data as a CSV file right from Excel, for example. Okay. And these are the libraries that we'll be using. Pandas, NumPy, which does a lot of numerical stuff, right? Matplotlib for some plotting. And the new one that we're going to introduce here, I think, is SciPy to do this linear regression. And we know how to do all this. So we're going to run the first cell to import Pandas as PED, NumPy, Matplot, and SciPy. It's just connecting for me up here in the corner. So as soon as it connects, it'll run that cell. And it gave me the check mark, so that's great. This is set up, as something that you could give to students. So it goes through a description in this theory block of the Bradford protein assay. It describes linear regression. I'm not going to go through this, but it lists what the variables are and how we're going to do it, what R squared means. And it does connect back here to things that we've looked at. It also then covers solving for unknown concentrations because that's what we use these curves for. We're going to pull this data directly from GitHub, but remember you could also upload the CSV file. You can click on the files, right, and just drag it into this space if you wanted. But we're going to get it from GitHub. And so we're going to pull that file in. And if I look over here, actually, I can open this back up. It appeared there. So I grabbed it straight from GitHub. And that's really nice because it just pulls it directly from GitHub for the students. So now what we're going to do is we want to load this data into a data frame. And so, so I just state the file name. I set the path, which is just content plus this file name. And then we're going to use that PD, this pandas read.csv. And lastly, we're going to just use the df.head, which Chris used, in order to see that there's stuff in that data frame. And it's pretty fast. It's got my data column names. I've got 3 replicates of absorbance values and the concentration in mg per mil for each of those. Right, we always want to look at our data to confirm it loaded correctly. Oh, I want to show you something kind of cool, I guess. If I click over here on that file, if I double click this, it'll display what's in those files for you, so you can see it over here on the right. I'll close that though for now. But yeah, so you can view that data. We're going to check for missing values because missing values can cause errors and clean that. So we're going to use the drop in A, and then we're going to look at the data frame. I didn't have any missing data, so it didn't really do anything. But what it did do is it also made a clean copy of this data frame into something that we now call clean data frame. So just moving that, the next stage in being that this is now clean data and we're ready to do our data analysis. Okay, so here's what we're going to do. We're going to calculate some new columns, really. So we're going to calculate mean absorbance and some standard deviation. We're going to plot that, add error bars, and do linear regression. And finally, calculate some unknown protein concentrations. So remember that we have each data point has 3 replicates. And so we're going to average that. And doing calculations in using pandas. So axis 1 is what we normally see, and it's calculating a value per column. When I do axis 1, it's actually going to calculate a value across the rows. So I'm going to at first define a list, right, of these replicate columns. They're just called rep 1, rep 2, and rep 3. And I know that because I just looked at it right here, rep 1, rep 2, rep 3. But we also looked, Chris showed us how we can get the names of the columns, right, by df.columns. Okay, so then to calculate that average across the row, we're going to create a new column called average, and that's going to just be equal to the replicate columns, right? And we're going to calculate the mean of those across the row. So that's what that axis 1 means. And then standard deviation, we're going to do something very different, very similar, but using the standard deviation. And finally, we're going to display our new data frame with these new columns. Okay, and you can see they've been added just that fast. And so we've got our average values for these, these are averaged, and there's our standard deviations. And so for our first exercise here that we're going to do is we're going to explore that standard curve. Okay. So we've got our values, and what we need to do is it says in the code below, print the average absorbance at the highest concentration. All right, so if I normally started writing that, it's going to it would use the AI to help us along, but we can just use the print command. So if I start typing print, it's still going to give us let us print whatever we want here. And let's see, it says we want to print our clean data average 5. I'm just going to copy this. So if I just do average, this is telling us it's in that 5th slot here. So I went ahead and clicked it, and you see that it printed 0.604 right here, and it printed all these decimals, and it's printing this number right here, which is 0, 1, 2, 3, 4, 5. So this is my highest average. Remember, we can also use the fprint if we wanted to do that. It asks us now to create a new column called CV%. And it tells us how to calculate that. So to create a new column right in here, so we're going to call that clean data frame. So, and what it's auto completing right now is not part of the AI. It's actually just looking at what variables we have already in our code. Clean data frame. We're going to put our brackets, and it wants us to call this CV underscore %. Okay. And then it tells us how to calculate that. So if we just copy this right here, well, we can run that. Okay, yeah. It says, oh, well, what is this standard deviation? So where is my standard deviation located, right? Well, we made a column called standard deviation, but it's clean data, right? It's this, right? So we're just going to grab this whole clean data frame standard deviation. So I'm going to paste that in there. And the average is the same thing. We're going to need to tell it where that average came from. I'm just going to paste that and change this to average. And I have to delete this. So now what we've got is we're creating a new column that's equal to the standard deviation of the data frame right divided by the average times 100. And thankfully I did that correctly. And you can see that it returned an infinity for 0. But we've got our CV%. If we had trouble doing this, somebody asked about, oh, are there answers? I've kind of embedded in mine. Paul noted that we do things differently. I embedded in mine these answers, so you can actually open this and see the answers. They just remain hidden, but you can click them open if you'd like to see them. Okay, so we did some cool things. We made a new column. We did some math. All right, so let's do some plotting. So let's look at our scatter plot before we do anything else. And so this is using matplotlib. So we imported it as PLT. This just dictates the size of the figure, what it's going to plot, okay? And it tells us if I mouse over this, all right, it's telling me plot X, Y, and then there's some format, all right? And so this is my X axis data. This is my Y axis data. This O data is telling us or telling me what it's going to use a circle for the points and what we want to label. I think I can still do this with, even though I've disabled things. If I highlight this right here and I click, I think I can click over here and on my little magic wand and click explain code. It will take a moment to think, but it's going to explain what this does. So it's telling me exactly, specified they should be displayed as circles and what the descriptive name for the series is in the plots legend. So this is a nice way that you can quickly identify what code is doing. Okay, so here we are. We're going to go ahead. This is going to make some X and Y labels, title it, and give a grid and generate a legend and show the plot. So let's go ahead, and do that. All right. So it generated that. Again, it's fun to just play with the code. If you want to see what happens when you change things, I mean, let's change this to 3 for my plot fig size. All right, so now it's much more, compacted, right? So it's got my X and Y values, 8 inches by 3 inches. So again, all these things, if you change one thing at a time and run the code, if it breaks, you can always change it back. But they can help you learn what they're doing. Okay, we had a question earlier about adding error bars. And so, right, so we can understand something about the consistency of our measurements. And so we're going to add the error bars as equal to the standard deviation, but you could create a different column that was something else that you could use for your error bars. And so that's really what we've got here. We're just adding this. And so let's go ahead. And you can see that it's called plot error bar, and we're still giving it the X and the Y. This is the error bar, and we're changing the formats. So we'll generate that graph. And we can see it put error bars on this. Again, let's play with the cap, which says is the size for the error bars. Well, if I change that to 10 and rerun that, they get much larger, right? They're wider, which is kind of cool. It's maybe a little easier to see than that this has the error bars here are much smaller than in this point for 0.3. Okay. So, all right. So we generated some points. We generated some error bars. Here's where I'm going to just digress for a little bit on line regress. So we're going to use this SciPy. All right, we imported it, and it is going to return a lot of these values: the slope, the intercept, the R value. We need to square it to get R squared, the P value, and the standard error. Okay. How do we figure out what it does and what it returns? And so how can we figure that out for ourselves? So I'm sharing my entire screen so that I can jump over here. But yeah, what I want to do is open up this document, okay, in a new window. Here's the documents for SciPy. And so I have to, say yes, go ahead and go there. And I've got for line regress. And so here's the parameters, right, that it needs to take. So X and Y values, right, 2 sets of measurements. It also tells me what it returns. So and here's those 5 things. Well, let's count them actually. So slope, intercept, R value, P value, standard error, and this intercept standard error. So it's actually 1, 2, 3, 4, 5. It's actually returning 6 things. We're only going to pull 5 since I didn't care about this, but it's going to return them in this order. Okay. So that's something that's important about how it's going to return that. And there's often examples of how to use it. All right. Notes on compatibility. Here's those examples. And so you can see this is not as good a data set as the one we had in terms of tightness, but you can see how you can use the line regress right here. And they're going to do it a little differently than we're going to do it. I'm going to show you both ways. How do we get all these parameters? That's really what we need to talk about, which is called unpacking. Because when we use this line regression, it's going to return at least 5, 6 values, but however many we want there. How do we get those? So what we can do is feed it all of these values. So the slope, intercept, these are variables. So the slope gets assigned the first one, the variable intercept gets assigned the second one, R value gets assigned the third one, and so on. So that's one way we can do that. The other way is to say that this result gets all of the parameters, and then result.slope is the slope, result.intercept is the intercept, and result.r value. And those are referred to in result by what SciPy says they're referred to. So slope, intercept, r value. Note that I can call, if I unpack them in this way, I can rename these things any way I want. That may be a little bit confusing, but hopefully we'll see how this works. Okay, so here's where we're actually going to do this. And we've got our concentrations, our average absorbances. And so again, we want the slope, intercept, R value, etc. We're going to feed it the concentrations and the average absorbance values. Okay, so we're going to click run, and it ran. It is going to take then that R value and square it. The asterisks, all right, is squaring it, this 2. And we're going to generate a line from that. Okay, so this is now using, NumPy to generate the lines, all right, from this. And we've got our slope that we need, and our line X, and our intercept. All right, and line X was defined here. So this defines our line that we're going to plot. Here we go, creating the figure size again. We're going to do it with error bars. We know our X and Y values. Here's our error bars. And then we're going to add that best fit line. Line X, line Y. And it's got that data again from using the slope and the intercept here from SciPy, right? Our stats.line regression. And this is what we generated. So we fit that best fit line right here. It's R squared is 1, and it gives us the equation of the line as well. All right. So, and again, you can do fun things here. Like I'll just do a fun one. Like if you wanted a pink line, you can type pink and rerun that. And then now your line is pink, for example. Again, we're controlling the number of decimal places that we're getting in these values here. And these are generating our labels. All right, so we were able to generate a Bradford assay with a best fit line, get the equation of that line. All right, we've got to do something with this now. So one of the things we might ask our students to do is, well, did you do a good job, right, at generating a standard curve? And so we can write some if-then statements. So for example, if their R squared value is greater than or equal to 0.98, we might say that's a great job. If it's a greater than or equal to 0.95, right, it might be an acceptable fit, pretty good, depending on what you're doing. And if it's less than that, you might say, well, maybe you've done something wrong. Okay, and so we want to print these regression statistics with labels, and we've got a hint from this first one. All right, I'm just gonna we've got this partially filled out. Printf slope equals. Well, we're just gonna grab this part right here and dump it in there. So we're going to use our curly bracket, and we're going to print the slope to 4 decimal places. And if I click run, that will certainly give us that slope. And if I go up here, there it is. There's our slope. So the intercept is going to be the same way. We can learn from this. So intercept is what we called it, colon dot for F. R squared. I forget what we called it, so I'm going to just go up and look again. Oh, we called it R underscore squared. So again, putting that in some curly brackets. Colon dot or F. P value, curly brackets, P underscore value. Colon dot for F and standard error, which again, I forget what we called that one. Standard underscore error. I'm just going to copy it. Colon dot for F. So if I run this again, you can see that I've got numbers. This p-value, though, is just listed as a bunch of zeros. So that's not super helpful. This little f, right, is giving me in decimals. But if I change this to e, it'll give me exponential notation. So now I can see that this value is actually 3.22 times 10 to the minus ninth. So that's understanding and learning what these are can be helpful. And again, you can find this in the Python documents, just like I looked through the SciPy documents. Okay. Okay, so now we can do this, write an if, if else in order to fit that. I've got time, so I think that's okay. So I'm just going to delete some of that. So if r squared equals 0.98, we wanted it to print excellent fit, right? So we can just say print I'm going to just copy this. Print excellent fit. Okay. And my r squared is, so that should actually work, and it does. Let's jump down and say else. I'm just going to jump down to else. And I'll say print check your data. And that shouldn't do anything right now unless I changed my r squared, change my r squared value, which we can do. I can just say this is r squared equals, we can set that variable to anything we want, 0.56. Oh, so now I've got to check my data. But we also wanted to do something in the middle, elif, because it was telling us to use if else. Elif r squared, so there's my r squared value, is greater than 0.95. Now, I want to share what's going to happen here. We need to print acceptable. Oh, let me put this, let me comment this out. So it's just, I already said it. So now it's not going to go back. So I'm going to put it back to 0.999. It says excellent. Oh, that says excellent fit. What if I put it at 0.96? Acceptable fit. Okay. And then if I'm at 0.94, check your data. All right. So we were able to write some code then that evaluates all those. We have an answer here then again, and our answer looks just like this one. So that's wonderful. Okay. Let's move on and do what we want our students to do, which is to calculate unknown protein concentrations using this. And we're going to, dictionaries were mentioned. I can't remember the detail behind them, but this is a dictionary. They store key and value pairs. So this is like a name and its absorbance. So this is again distinct from a data frame. We can do this with a data frame and we will, but I wanted to show you how to do this with dictionaries. So we can create a dictionary of like cell-free extract, and this would be its absorbance value. The flow through elution 1, elution 2. So if we've got stuff coming off a column. And this is how you iterate through that. So for the key comma value in this is just a for loop. It's going to iterate over all these, and it can then calculate the protein concentration using the value for our absorbance minus the intercept divided by the slope. So it's using our equation rearranged, right, that we needed from above. So it's going to do this calculation for all of these. And so we've got these concentrations now that it returns in mgs per mil. And so again, that's really just rearranging Beer's law. Okay. I'm sorry, not Beer's law in this case. Sorry, it's rearranging our equation for the line. Okay. We can do that same thing with a data frame. And Paul mentioned that we could use zip, which is like a super cool feature. So here's our samples. And here's our absorbance values. The thing is that has to be true is that they have to line up, right? The first item in this list has to be the first item in this list. Second has to go with the second, et cetera, et cetera. And we can use this zip command. So this list zip will take the samples and their absorbance, and it's going to then drop them into this data frame. So we can look at that, and it's actually really cool. So now it made a data frame, and we went ahead and did the calculation on that data frame, which if I just did sample underscore df here, we can see it in between. Oh, no, I didn't do that. I think as it overwrote it. I'm going to throw some commas in to see what it looks like before. Yeah. So again, if we stop right here, we've zipped together those two things, have our sample and sample absorbance. So then if I uncomment these, this is just again using the equation of our line. Oops, I'm going to need to put the one back on this one. To add that new column where it calculated, and you should be able to see that these values are the same values that we returned up here. So that's two different ways of solving a problem. So what we can do then is if you wanted to, we've got like 5 minutes or something. The idea would be, well, okay, can we have you guys do this? Can we add another unknown here for wash 1? And wash 1 is going to have an absorbance of 0.357. And we can do that using both the dictionary or the data frame. And so I will wait for 2 minutes. I don't think it takes very long to do that before I start typing. I'll go ahead and do that. W1 colon 357. That's it. That's all I had to do there. So I can run that, and it's now added W1, and now it's done that calculation because it was iterating over the list, however long it is. We can do the same thing then to the data frame. We're just going to need to put a comma, give it that value, W1, and I need to add it again here at the end, 0.357. And we can see it came up with the same 0.295. Now, if I didn't put this in the right order, if I decided to add one at the end and one at the beginning, it's going to make those assignments incorrectly, right? So that's the thing about zipping those together. There has to be an order. Okay. So in this, there's some wrap up stuff here, but we've got a workflow that takes you or some students through how to calculate the sample absorbance, how to interpret their R, their R square value. I've got some optional challenge questions, and I think this challenge question 1 is like my favorite. It's correcting for sample dilution because nobody's CFE is going to be really that close to their, their elutions or whatever. So, what this is assuming is now that we've made a dilution factor for each of these. And so we've got some starting code here, and it's going to take these samples and zip them together to make a data frame, and it can calculate those. But it says here, create a dictionary of dilution factors. So, right, they've got to be, the key would be the sample name and the value would be the dilution factor. We would have to add those pairs here and then correct for those. So that's something cool that we can do at the end then. We're just about out of time, and I think I started early anyway, so I should stop. But this is how we can add those dilution factors. And so I'm just going to copy that right out of there. Oh, I don't need to copy all of that. And I'll click run. And now you can see, oh, well, that didn't work right. Oh, because sorry, I didn't need to replace more code. My apologies. And sample DF. Let's see what this one runs. There's the dilution factor has been added, and sample concentration equals this. And so now you can see that all of these have been multiplied, right, by the correct dilution factor. And that's something that students would have to do realistically, really, in order to get it in the linear range. There are other problems. They all have answers down here, but you've stuck in there for a long day. So I'm going to turn it back over to Paul, and I'll answer any questions that may come up in the chat bot. But I hope you've enjoyed kind of seeing what a notebook might look like for students where they can tweak a little bit. They could import their own data into something like this and learn a little bit of coding. Thank you.
Well, thanks everybody for staying with us. Let me share my screen. And just wanted to go through a couple of things here. First of all, here are some links, and you'll have access to this slide deck. And you can email us. Originally, I had codingbmb@gmail.com. It's actually codingforbmb@gmail.com. That's so some people got an error on that. So you can reach out to us at the codingforbmb@gmail.com if you want to join the Slack channel, want to get into the LinkedIn group or whatever. And we'll be happy to set that up and welcome you on board. We do have a couple of workshops coming up. On the 19th and the 21st of May from 1 to 4 in the afternoon, we're going to do something similar to today, but with more depth, more length. So it'll be actually 2 3-hour sessions. Those are Eastern times. And you can find out about that by going to the PyBMB website, which I've shared that link in the chat room. Also, at the Biennial Conference for Chemical Education, July 26th through 30th, we're going to be giving a day-long workshop there. We don't know the details exactly which day it's going to be on, but once we have that information, we'll post it on the website as well. And just to give you a feeling for our 3-year plan. So right now, this summer, we're doing our introduction to coding, and today's session was an abbreviated version of that. That's happening now. And then we're going to do a more detailed coding for BMB where we go through 5 or 6 different applications that are pretty common and learn some new skills. And then that's going to be in the fall. And then the spring of 2027, we're going to look at teaching with coding. So talk about live coding, basically what we did today. And then we're going to do the same sequence of workshops as the second offering the following year. So next summer, we'll do this one. In the fall of 2027, we'll do the second series. And then the spring of 2028, we'll do the third series. So those are opportunities if you want to learn more, be part of it. I'd like to acknowledge some folks. I'd like to acknowledge, first of all, NSF for providing the funding for this effort. Our grant is called Collaborative Research Promoting Computational Literacy in Biochemistry and Molecular Biology Education. Thank that the people at the Protein Bank at Rutgers, Protein Data Bank in Rutgers, also the Molecular Sciences Software Institute. That's where I learned everything I know. NSF, and then our 4 universities, Penn State, RIT, Wabash, and James Madison.